the microbes coding for functionality become a very critical aspect of how we work and function as a species. And in fact, you compare that to other species that have not incorporated a large mass of microbes in their system, right? So we look at lower order animals like the poor earthworm. Even though they have that earthworm has more genetic capability than we do innately, the earthworm also doesn't have 2.5 million microbial genes, right?
Hi, I'm Dr. Ted Achacoso, your host for this podcast. Today, we have a very special guest who is no stranger to the world of health and wellness. He is a research microbiologist, a trailblazer in the field of probiotics and gut health, and a passionate advocate for science-based natural health solutions. I am thrilled to introduce my dear friend, Dr. Kiran Krishnan. With his extensive knowledge and groundbreaking research, Dr. Krishnan has been revolutionizing our understanding of the microbiome and its profound impact on overall health. Via his innovative approach to probiotics, Dr. Krishnan has been pushing the boundaries of what we know about our bodies and how we can take control of our health. Among today's $64,000 questions that Dr. Krishnan will answer are: number one, what are the most exciting clinical developments in the field of gut microbiota and probiotic supplementation? For example, the links of gut microbiota to a wide range of health conditions, including obesity, diabetes, allergies, acne, and autoimmune diseases, as well as to lifelong health. Number two, how do outer membrane vesicles violate the dogma that bacteria and human cells should not communicate directly? Number three, why is the gut microbiota now considered an organ in the body that is postnatal, or an organ that grows right after birth? Whether you're a health enthusiast, a curious learner, or someone who is looking for natural ways to improve your well-being, you're in for a treat today. So sit back, relax, and let's deep dive into the fascinating world of microbiology with Dr. Kiran Krishnan. Hi, I am Dr. Ted Achacoso, your host at the Smarter Not Harder podcast. My guest today is Dr. Kiran Krishnan. He is the most nice gut microbiota ecosystem engineer you'll ever find, and also the most knowledgeable. Kiran and I have traveled together. I've invited him to the Philippines to lecture a few times, and we've traveled to San Sebastian, right? And to the south of France, and we were both lecturing to key opinion leaders on gut microbiota. So he looks so much younger than I do. Don't let his looks fool you. It's his gut microbiota that's actually keeping him young, right? Hi, Kiran.
Hi, Ted. It's so nice to be with you. I'm so glad we finally get to do this.
Yes, yes. It only takes you to be out of your corporate role now.
It took me to retire before we could do it.
Yeah, let's talk about that later. I would love to learn about that, the successful trajectory. It's actually a very, it's something that actually I was looking at, your style, your strategy, etc. It's just been totally well executed. So folks, that does make him not only the world's best gut microbiota ecosystem engineer, he is also a fantastic entrepreneur. He just did a great exit from his big company and is now looking, I think I heard, to do another one. So
yes, a couple of them. I'm trying to follow in your footsteps. But I've always said when I grow up, I want to be Ted. So,
but I want to follow in your footsteps in terms of the success, right? So
yes. Well, see, you tend to have a habit of building companies and giving it away.
Yeah, I do.
I just changed the ending where I sold it instead of giving it.
That's so true. You know my history pretty well. Kiran and I had fun introducing, his probiotic products in Asia when, I used to consult for a large company in the Philippines, and they had a Southeast Asian distribution. And Kieran and I sort of like engineered that whole thing. And it is actually quite interesting because Kieran was talking to like the mid-level. I said, no, let me make you talk to the owners.
Yeah.
So,
That was much more effective than yeah.
it's a lot faster,
Yeah.
right? So I would like to dazzle our audience now with
Yeah.
your brilliance, Kiran. And I have actually put a list of the 5 most important developments in the field of gut and gut microbiota science and technology in the past 20 years.
Yeah.
I'll enumerate them, and
Okay.
then I'll ask questions. I'll repeat them and then ask questions in each of them. Okay?
Excellent.
So here are the 5 most important developments in the field of gut and gut microbiota science and technology. Number 1, the gut microbiota is now considered as an organ in the body. In fact, it has been dubbed as the forgotten organ. Number 2, because the gut microbiota is now considered an organ of the body, previous bipartite axis, such as gut-brain axis, is now called enteric microbiota gut-brain axis to reflect this tripartite axis. The third, the gut has an enteric nervous system that is autonomous of the first brain. Thus, it is now called the second brain. In addition to this finding, the gut is also now considered to be the largest immune system of the body. Number 4, one of the biggest news is that of forbidden interkingdom communication. There is evidence that outer membrane vesicles, or OMVs, can fuse with human cells, including intestinal cells, and deliver their contents to the host cell. These include small RNAs, which can influence the function of the host cell. And number 5, and we'll dwell a little bit longer on these because this is a series of questions, the development of clinical tests for gut microbiota, for example, stool and urine tests for bacterial and fungal metabolites and DNA, the links of gut microbiota to a wide range of health conditions, including obesity, diabetes, allergies, and autoimmune diseases, as well as to lifelong health. And of course, your specialty, the development of interventions that can be used to modulate gut microbiota, such as probiotics, prebiotics, and fecal microbiota transplantation. Okay, so let's take the first one. The gut microbiota is now considered an organ in the body. So, Kiran, can you describe for us the gut microbiota ecosystem and its functions?
Yeah, absolutely. So, and I think it's absolutely is a well-defined organ now. When you think about the gut microbiota in the sitting there in your digestive tract, number one, there's a massive surface area in the digestive tract, right? And I think people may not have that understanding at the forefront of their head. When you unravel the entire digestive tract, it has the total surface area of at least a professional tennis court, right? Which is a massive, massive piece of land that's all folded up into your digestive tract. Now, every square millimeter of that is covered with microbes. Something like 30 to 40 trillion microbes reside in that area. And those microbes are responsible for so many things. Now, before jumping into what they're responsible for and what they do, just to put it in context, right? We've known that our genes are a very important part of how we function and what we do and what we can do and so on. But humans are wholly inadequate when it comes to our own genetics, right? We have about 22,000 functional genes. You compare that to an earthworm that has about 32,000 functional genes, right? We're not that cool.
No.
When you think about it, we're really not that sophisticated. But then you think about, well, why is it that we can do the things we do and the way we function? We have over 2.5 million microbial genes in that tennis court, right? And that actually serves as the largest source of capability in the human body. And so the microbes coding for functionality become a very critical aspect of how we work and function as a species. And in fact, you compare that to other species that have not incorporated a large mass of microbes in their system, right? So look at lower order animals like the poor earthworm. Even though that earthworm has more genetic capability than we do innately, the earthworm also doesn't have 2.5 million microbial genes, right? So it'll stay an earthworm. It'll stay burrowed in the ground. It won't come up with technology and things like we've been able to do.
Mm-hmm.
So that's the context of the
Unless
gut
it's a
microbiota.
tapeworm, it will
Yeah, well, that's true.
it will
And
come
then it'll
up. It
find
will come
us.
or
And
Ascaris lumbricoides right up your intestine.
exactly. And it knows where to go, where the rich capabilities are, right? Uh, but it's absolutely fascinating when you break it down that way and you think about it, right? Think
Mm-hmm.
about the surface area, the number of microbes, the number of genes that are present in this gut microbiota area. Now, then, when we start thinking about, well, what is it responsible for?
Mm-hmm.
It's almost easier to try to understand what it's not responsible for, because it's responsible for more things than it's not,
Mm-hmm.
right? So some of the functionality that we know of, like for example, coding for our eye color, skin color, height, things like that, we have genetic material that codes for that,
Yeah, those are
right?
structural genes, right?
Yeah,
Those are
those
structural.
structural genes, we have
Anatomic.
those innately. Yep.
Yeah.
The outside of that, though, when we think about day-to-day function, when we think about simple biochemistry of producing compounds, metabolizing things, excreting things, breaking down the
Mm-hmm.
material that we put into our system as food, and then assimilating those into more useful ingredients, all of
Mm-hmm.
those things, the microbiota is responsible for in one way or the other, whether it's directly and only responsible for it, like in the case of short chain fatty acids, right? We know
Mm-hmm.
butyrate is critically important for
Oh
all
yeah.
kinds of immune functions, metabolic functions, and all that,
Yeah,
right?
and remember, low butyrate levels are associated with higher incidence of colon cancer, right?
Exactly. Yep.
Yeah,
Colorectal cancer,
yeah,
Crohn's, colitis, microcolitis, all of
yeah.
those conditions, and
Mm-hmm.
also most things under the metabolic syndrome category, right? So
Mm-hmm.
diabetes, cardiovascular disease, obesity, they're all low butyrate, low short chain fatty acid conditions.
Mm-hmm.
And the thing is, we can't produce short chain fatty acids ourselves, right?
Right,
We can't manufacture that through our own cells. We
right.
count on the microbes to do that for us. So
A-
it's
And
just
also vitamin B12, right?
exactly, yeah, B12, right?
Yeah, yeah,
Exactly.
yeah,
An essential vitamin that we need to
yeah.
function as part of our basic cellular mechanisms.
Mm-hmm.
We can't do that ourselves. Another one that I really love as a great example of that is the importance of urolithins, right? And
Mm-hmm.
urolithins for managing our mitochondria and the
Mm-hmm.
function of our
Mm-hmm.
mitochondria, right? Turning
They're
over these
markers.
mitochondria,
Yeah,
these, yeah, important
yeah,
markers. And
yeah.
we know the mitochondria are the engines of our cells, and if those engines die, the cells die, and we age and we get sick and so on. And so
Mm-hmm.
maintenance of those engines becomes really important.
Right.
And this compound urolithin is so critical in maintenance of those engines. We can't make urolithins ourselves.
Yeah,
Microbes make it
I
in the
love
gut.
the way you pronounce it. When I lecture, I say urolithin. When I
Yeah.
lecture in the mitochondria, but urolithin sounds more exotic. So
It does. Your license sounds like what an academic would say. So,
no, because a lith sounds like a stone, right? An
right.
otolith
Oh,
or
yes.
something like that.
That's
That's
right.
why I say lithin, because it's like, okay, that's just they're not stones. But
Right.
for those listeners who are not familiar with urolithins, there are usually a common pathway, especially in mitochondria, that marks a mitochondrion for mitophagy, right? So
Yep. Exactly.
it's gonna eat up the mitochondria so that you can maintain a stable population of mitochondria.
Yeah, and for the audience that aren't familiar with what happens with mitochondria, right? So mitochondria being the engines of the cell, like any engine in your car, over time they get worn down.
Mm-hmm.
They get worn down by just functionality, by producing these reactive oxygen species as a byproduct of making energy,
Mm-hmm.
and those biological engines get worn out and need to be replaced. And
Right.
if they don't get replaced, then your cells become accumulated with dysfunctional engines and they can't function properly. There was a fascinating study, pathology study, where they took tissue samples from a 5-year old and a
Mm-hmm.
90-year-old and sent them blinded to pathologists to grade the tissues and try to distinguish what was different about them. And the only thing that they could distinguish between the two sets of tissues was that the 5-year-old's tissue had 100% functioning mitochondria. The 90-year-old's tissue had more than 95% dysfunctional mitochondria, right? Meaning
Yeah.
broken down engines. Not all the cylinders are firing, right?
Well, Douglas Wallace had study, right? And he stated this in one of his lectures, which was so disappointing for me. He says that when you're in your 40s, like 60% of your mitochondria are already mutated. It's like
Right.
it's all downhill for you
Yeah.
from there on. It's just like holy fuck. But
Right.
anyway, it's just like the thing that actually was Kieran, that fascinates me about this is that I consider mitochondria as endosymbionts, right? They live
Yeah.
inside our cells, but they are bacteria. And then
There.
I consider gut microbiota as exosymbionts. They
Yeah.
are bacteria, but live outside our cells, but they're all part of us, right? And the
Totally.
interesting for me is that they both come from the mom, right?
Yep.
They
Yeah.
both come from the mother. So the mother says, "Let's dirty it up,"
Yeah.
make this new host, this human body, right, very resilient to
Yep.
a lot of oncoming onslaught
Yeah.
of other bacteria that's coming from the environment.
Yeah,
Uh,
no, and what you said is actually a really elegant thing for people to keep in mind because you think about the endosymbiont mitochondria, right?
mhm.
That thing had to occur for humans to even exist, right?
Yeah,
The
right.
eukaryotic cell wouldn't exist if this old ancient pleiotropic bacteria did not come together with an
Yeah,
archaea or whatever it was
yeah.
to form what is now a human cell. Now, for that structure to function properly, we still need microbes as exosymbionts to dictate the function and the turnover of that component. And this was probably happening predating humans,
Mm-hmm.
right? So the ancient mitochondria that existed on its own as an individual cell, if you will,
Mm-hmm.
probably utilized things like urolithins or urolithins
Mm-hmm.
in the environment to turn over or to induce functionality. We've just now benefited from all of that interesting biological evolution, right? And knowing that becomes really important because we have to support all these systems.
Yeah, there's an interesting thing that I read before, tidbit I read before in the coming together, in fact, of the mitochondria and the anaerobic cell, right? And it is said that the kleptogenes, the mitochondrion actually had kleptogenes,
Yeah,
right? Which is
yeah,
now been transferred to the nucleus, of course.
yep,
But it already was producing ATP by itself using
yeah,
oxygen, but at that time the oxygen concentrations were fluctuating,
yep,
so could not function if there was no oxygen, and therefore it had genes that would actually steal energy from the archaea,
yeah.
from the anaerobic organism, which is interesting. Yeah, people think that, oh, they went into cooperation. No, 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 this is a matter of survival. Right,
Yeah,
right.
and
And
one
that's
got
why
kidnapped.
and that's why we describe the gut microbiota as an ecosystem, right? It's
Yeah.
these organisms are in competition and cooperation and compromise with each other. It's funny. I was investigating a small model, AI model, right? A generative
Mm-hmm.
model for AI, and I asked it, "What are the three C's of evolution?"
Mm-hmm.
And I tried it in a model called Orca, and it was very funny. He said, the three C's of evolution are competition, cooperation, and selection. And I said, but selection is not a C. And he said, I
Right.
insist that the third C is selection. But but it was really funny, right?
That's
It's like,
awesome.
oh my God, this AI language model is arguing with me. But anyway, just looking at what you said, there was a very interesting figure, Kiran, that a diagram that I saw, which actually really summarized what you said, where with all of these functions that are actually subsumed by the microbiota, right? So basically, they pre-process all of our food and everything that comes
Yep.
into your gastrointestinal tract, including drugs, right?
Yep.
There was a study where the oral cancer drug wouldn't function unless there was actually two species of bacteria that were present, right?
Yep.
So they pre-process, and then so they meet up your food or whatever you put in there, food and drugs or whatever you're putting in there. I don't know, you kinky ones, but and then there is the microbiota to the gut lumen, right? The gut
Yeah.
itself is a separate organ. That interaction between the gut, the intestinal epithelium, and the microbiota itself, that's the second interface. And the third one is the metabolites, right? That's
Yep.
produced and absorbed. So it's nice to view it that way, in terms of being able to see how extensive really the role of gut microbiota are, right?
Yeah, absolutely. And I think, another way to view it is like if you think about from human to human, we're so unique and different, right?
Yeah,
In terms of how we respond to things, how we respond to diet, food, drugs,
yeah,
environment, and all that. And yet, genetically, we're almost identical,
yeah.
right?
Hmm.
And so, from a gene perspective, we're basically exactly the same. And yet, a lot of our uniqueness comes from the unique microbiotas that we have. So that
Yeah.
alone kind of helps illustrate everything that the microbiota sitting in your gut is dictating for you on a day-to-day basis.
By the way, I just like to clarify for our audience too that microbiota, although we're referring mostly to bacteria, they actually include some yeast cells in there. They
Hmm.
include some viruses, right?
Sure.
So it's basically an ecosystem of organisms that are in that. So so let's go to the second one. It
Yeah.
said because gut microbiota is now considered as an organ in the body, the previous bipartite axis, such as the gut-brain axis, is now called the enteric microbiota gut-brain axis to reflect this tripartite axis. So, how do the gut microbiota affect other organs? For example, the brain.
Yeah. And there's a lot of exciting stuff happening in the gut-brain
Mm,
axis world,
mm,
right? So if we think about the connections, the physical connections itself, then we have, of course, the enteric nervous system
mm-hmm.
of the digestive tract, which is connected to the brain through the vagus nerve.
Vagus
So
nerve,
we've got
right?
a physical connection there from the two neurological systems. And then we have the circulatory system, of course, which can carry nutrients and compounds that are made in the gut or assimilated in the gut to the brain itself. And then we also have the immune system.
Mm-hmm.
The immune system, which uses both the circulatory system and can use the lymphatic system to make its way to the brain. We know that somewhere around 75–80% of the immune tissue in the body exists in the gut. The microbiota in the gut influences the immune system quite significantly. And then the immune system plays a very important role in how the brain functions,
Mm-hmm.
especially when we look at things like stress response, right? Because
Mm-hmm.
we know that microglia cells, which are these macrophages, if you will, within the central nervous system in the brain, play a very important role in upregulating sympathetic activation and blood perfusion and using immune inflammatory triggers and so on. And
Mm-hmm.
then the activation of the HPA axis, the hypothalamic pituitary adrenal axis, so to put you in that flight or fight response or bring you down from the flight or fight response, cortisol response, all of those things are dictated by immunological responses in the brain, a lot of which start and stem from the gut. So, and then another very important component of all of this is we've always thought of these neurotransmitters as
Mm-hmm.
being brain and central nervous system
Mm-hmm.
sole compounds, right? Now it's pretty clear that virtually every neurotransmitter discovered is also found in the enteric nervous system. So plays a role not only in the gut, but maybe even is controlled to a certain degree in the gut. We know serotonin and dopamine are made in the gut, right? We know things like brain-derived neurotropic factor and other components that impact the
Mm-hmm.
brain are manufactured in the gut and controlled in the gut. So I always think of the gut and brain
Mm-hmm.
actually is not as two separate systems that are connected. I tend to think of them as two parts of the same system. You
Right.
know, they function so similarly and in such concert with one another under the right conditions. Of course, there's lots of people who have quite a disjointed gut brain. And so, and there's a lot of symptoms of that, right? But they're really two parts of the same elegant system.
Yeah, actually, when you look at it in terms of embryology, right,
Hmm.
you see that the gut is forming there, actually, from the endodermal tissue, and then
Yep.
you could see the brain actually is forming from the ectodermal tissue. They
Yep.
actually are forming in different ways, and they're functioning autonomously. They are, when you have a gut feeling about something, you trust something like that,
Yep.
and as we were talking about earlier, one of the things that's on fast track for approval in the United States, at least, is the use of MDMA for PTSD, right?
Yep.
And uh, uh, you know, 90% of the serotonin in the body is produced in the gut, and that's why an oral MDMA, for example, will produce some gut discomfort.
Yep.
They call it the serotonin gut, right? When they administer this in the clinical trials. So it's actually quite interesting how. Oh, this is again processing a drug or a potential drug that's about to, it's already approved in Australia for PTSD, right? And so you could see here that it's actually raising a lot more of the serotonin,
Yeah.
right? In there as it's an unusual amphetamine. It does in that it has a very low Ki value for cert, right?
Yeah.
So
Exactly.
Kiran, what are the have you seen any later studies now that have to do with manipulation of gut microbiota for depression and anxiety?
Yeah, we've been doing actually a lot of work on psychobiotics, and the
Mm-hmm.
idea behind psychobiotics are that these are microbes that reside in the gut or function in the gut that have a specific impact on the brain.
Mm-hmm.
One in particular that we've done a lot of work with at Microbiome Labs and then at our partner Novozymes as well is a Bifidobacterium longum, and it's the species number is 1714. Now, what's unique about this microbe is that it contains a number of carbohydrates. So, on its outer membrane, it has this carbohydrate structure, which we call an exopolysaccharide. So it's this like complex polysaccharide mix. So it's basically all these carbohydrate tentacles all over, if you will, on the
Mm-hmm.
outside of it. And then also within the cellular membrane, it contains a specific carbohydrate called peptidoglycan.
Mm-hmm.
Now, here's what's so interesting about how this functions. When you consume this probiotic bacteria, right now, there are certain percentage of people that have this bacteria natively in their gut already.
Are
But
those the normally happy people?
are normally happy. That's exactly, and that's how it was found actually. So there was a study done by the APC, which is one of the preeminent microbiome research institutes in Cork, Ireland. They were looking at older Irish women that had very well balanced moods, slept well all their lives, didn't have stress or anxiety and so on, and they were comparing their gut microbiomes to age match individuals that had lots of history of sleep issues and anxiety, stress, depression, and so on. And
Mm-hmm.
the main thing that they found was the presence of this organism at high levels in those really well balanced individuals. So then
Mm-hmm.
they isolated the organism and they said, okay, let's look at how this organism impacts mood.
Mm-hmm.
And then they found that this organism's unique in that it has this carbohydrate structure, it has a peptidoglycan within the cell membrane. Then they started doing studies. We were starting to look at how does that peptidoglycan impact the brain and impact
Mm-hmm.
mood. And this is where it becomes super interesting, right? So here's what we found essentially. When you consume a bacteria like this, or if you have it natively, what tends to happen is a microbe, when it gets to the terminal end of the small intestine, in that area called the Peyer's patches, which is lots of immune tissue
Mm-hmm.
there,
Mm-hmm.
right? There are actually dendritic cells. These are these antigen presenting cells in your
Mm-hmm.
immune system that can reach across the lining of the gut. They will
Mm-hmm.
grab these bacteria, they will pull them across, and they will consume them, right? So it's called phagocytosis.
Mm-hmm.
Now, normally, when dendritic cells or macrophages, these antigen presenting cells, are consuming a microbe, they're doing it to present antigens of the microbe to the rest of the immune system, right?
Yeah,
So, right?
let me chop you up and present you, right?
Exactly. Chop
Yeah.
you up and show the T cells and B
Yeah.
cells what's
Yeah.
here so that they can mount an immune response against whatever it's consumed. It'll do that with viruses, it'll do that with bacteria and so on. In this case, when it consumes this bacteria, it doesn't actually present any antigens to the rest of the immune system. It
Mm.
digests the bacteria on the inside, and then it spits out these unique carbohydrates from the bacteria into our circulation,
Oh wow.
right? So our dendritic cells are doing that for us. Now, these carbohydrates,
It's just a murderer, huh? And
yeah, it's just a murder.
it's
Yep.
basically eviscerating the bacteria just to get to its contents. Okay.
Just to get to its contents, right?
Yeah.
And then releasing the contents to the host. All the while doing this without presenting any antigens. And that's really important because that means that these microbes are non-immunogenic, meaning they don't
Mm-hmm.
elicit an immune response in the
Mm-hmm.
body. And the dendritic cells have somehow distinguished between these microbes that it wants to consume, break down, and squirt out the components versus microbes that it normally consumes, breaks down, and just presents components to
Wow.
activate the rest of the immune tissue, right? So the dendritic cells somehow know that this type of bacteria is important to bring across
Mm-hmm.
to the blood supply, right?
Right.
And I'll talk
It's
about
in the mafia payroll.
yeah,
Okay.
exactly. Yeah, that's essentially it, right? It's figured this out somehow.
Yeah.
Now where this actually originates, and this is where it becomes fascinating, is you see this happening in pregnancy in utero.
Mm-hmm.
So there's good evidence now showing that dendritic cells will reach across the mother's gut lining and grab certain bacteria and bring it across the lining and then take it to placenta and put it through the cord blood so that the baby gets exposed to certain types of bacteria, right?
Mm-hmm.
These types of bacteria, these psychobiotic bacteria, are one of the classes of microbes that dendritic cells do this to now, and this is when the baby's developing. Now, the reason it does it is, as it turns out, the placenta has receptors and carriers for bacterial carbohydrates, especially bacterial peptidoglycan, which is housed in the lining of these microbes, right? Now, when these receptors bind the bacterial
Mm-hmm.
peptidoglycan and transport them to the baby's brain has receptors for bacterial peptidoglycan. And when the baby's brain binds the peptidoglycan, it starts to initiate synaptogenesis in the
Mm-hmm.
baby's brain. It starts to initiate the formation of the blood-brain barrier. It starts to drive cellular differentiation so that you can form all the different parts of the brain. It
Mm-hmm.
starts to stimulate myelination of the
Mm-hmm.
central nervous system, right? So the putting the insulation on the nerves. So arguably, some of the most important things we want to happen
Mm-hmm.
in our growing child in the womb, which is the development of the brain and the central nervous system, we've outsourced that to this bacterial byproduct, right? Which is
well,
absolutely fascinating.
well, I hope it also drives the formation of neural networks for dirty thoughts. I'm
Right.
kidding.
I'm sure it does. It depends on how dirty the bacteria are to begin with, right?
Because also that's a man that's a source of creativity.
Yeah,
So
exactly. So these bacterial carbohydrates were so important from day one when your brain was being developed. Now, for the rest of your life, as it turns out, these are protective of the brain
Mhm,
against things like inflammation and overt activation of the HPA axis.
mhm.
These carbohydrates, when the dendritic cells bring them across and release it into the central nervous system, it's these carbohydrates that start to dampen the inflammatory response that triggers HPA activation and puts us
I
into the
see.
sympathetic state, right? So we
Yeah.
have studies showing that when you're exposed to these types of bacteria and the resulting carbohydrates,
Mhm.
that you actually start shifting away from the sympathetic back towards the parasympathetic.
Sympathetic. Yeah.
And it also changes brainwave activity. It actually takes you out of those high frequency brain waves like the beta
Mhm.
waves and actually puts you in theta waves and alpha waves more readily, right? So it completely changes perception of
So
stress and anxiety.
so these are also not only the happy bacteria, but they are also the kamikaze equanimity bacteria. Is that
Yeah, exactly. They are. They're the yogis, if
the
you will,
yogi,
also, right?
the yogis of offering themselves for sacrifice, right?
They are, yeah. I mean,
Yeah.
just think about it, right? They come in, they sacrifice their bodies, and within their bodies, there are very important components to maintain balance in our brain.
This is fantastic, Kiran. It's the first time I've heard of this. Can you take these peptidoglycans directly? Can you
You
consume them
can.
directly?
You can, yeah. It's just hard to produce them in high yield.
Mm,
So getting them from the microbes becomes a really easy way to do it. Now, one
mhm,
of the experiments we were doing was, can we enhance the release of the peptidoglycan by, for example, combining the bacteria with a enzyme,
mhm,
right? A lytic enzyme that would break up the microbes even more. Initial studies on that in animal models showed that it can enhance it, but then you may lose the recognition by the dendritic cells, right? Because that's a
Oh,
very critical
right,
component,
right,
right?
right.
The dendritic cells need to be able to see the
See,
microbe, recognize it, bring it, grab it, and bring it across, and that
mhm.
becomes the biggest bioavailable way of
I
getting it to the
see.
body.
I see. So, you just touched on really one of the issues. I'm sure you've worked on this on your career. Is that if you intervene with something, there's something
Yeah,
in evolution that may break, right? Like this
totally.
time, it's like oh, if we give this, then it will remove the recognition
Yep.
a portion or it's kind of like giving dopamine directly, and it basically decreases the number of dopamine receptors, right? It's just
Right.
like there is this basically payback
Yeah.
for what you're gonna do, and you just have to weigh whether or not it's worth the price, right?
Exactly. Yeah.
So
Yep,
so
and that's the struggle of a lot of prescription approaches, right? Because
hmm,
more often than not, prescription approaches are intervening in normal processes, right? And
right.
then you throw off the balance somewhere else. The benefit of thinking about things from a microbiome engineering perspective is if you can facilitate
Hmm,
the balance in the right way, then you shouldn't have those auxiliary dysfunctions, right?
yeah,
Because the system is always a system always has a tendency to stabilize, right? Because it is an ecosystem, and any
right,
ecosystem in the world wants to
right.
reach a degree of stability,
That's
right?
right,
And the stability comes from interactions, comes from competition, all of those good things. So if you can play within that space and not disrupt the natural order of how an ecosystem functions, then you can push the ecosystem in a direction without breaking it,
right,
and often we break systems in
right.
biologically by just going too hard, right?
Yeah, I've said this story quite a few times, but Kiran, I started this nonprofit, right? Health
Mm-hmm.
optimization medicine and practice, which teaches doctors and health practitioners, non-physician health practitioners, how to read metabolite levels and
Yep.
how to balance them, right?
Yep.
Without looking at the disease, etc. We just take
Totally.
a look at the optimal levels of metabolites. And when I was starting this, it's now 13 years old, right? I was starting this 13 years ago. I get calls from doctors like, Achacoso, how is your hormones, nutrients, and cofactors going to affect my drugs
Right.
that I'm giving this patient? And I go, this actually the opposite question. Your drugs have never been seen by the body in evolution, but
Right.
my nutrients, cofactors, and hormones, they've been seen and used by the body in evolution. So you tell me how
Right.
your drugs are actually going to affect the levels of my nutrients, hormones. And they were so aghast that was my response. But that's
That's
exactly the point.
fantastic.
That's exactly the point that you're making, right? It's
Yeah.
like the
Yep.
body has seen this before, and before we actually shove in the antibiotic in there for an acute infection, we should already know what it's going to do, right?
Yep.
We have already seen studies of antibiotics being given to kids. They alter their gut microbiota so permanently that
Yep.
in like 10, 13 years later, they become obese. And the only factor that could really isolate was the one-time bombing of their
Yep.
microbiota, right?
That's exactly right. Yeah, and it behooves us to, number one, respect the elegance of biology and the system that we
Mhm.
have, right? And I think that's one of the falters of modern allopathic medicine is there's a hubris
Yes.
that doesn't respect the system. It's almost like we can outsmart the body, right?
Yeah.
And we know better. We can intervene here. We can intervene there without really respecting the balance and the control systems in the body that already exist. That
Right.
a lot of it predates us because a lot of it comes from microbes and how
Right.
they interact with each other, right?
Yeah.
Given every one of our cells are essentially microbes in their own right.
Right. And in fact, in health optimization medicine and practice, if people are interested, it's at homehope.org. There is actually a module there teaching about evolutionary medicine,
Yeah,
where, in evolutionary medicine, you're actually looking at signs and symptoms as a defense mechanism of the body
yeah,
rather than a sign and symptom of disease. Like if you have this, then you have this disease. No, no, no. If you have this, then the body is actually trying to right itself, right?
yep,
By evolutionary means like pain, swelling, redness. These are
yeah,
all not signs and symptoms of disease, but the body's defense mechanisms and the body's way of healing itself. It's a very, very different perspective, right? And,
Absolutely. It really is.
and to your second point, it's like we're made up of organisms. There's a term called holobiont, right? Which
Yep.
I have pushed to the public because we're after holobiontology, right? So
Yep.
our mitochondria are bacteria. You have gut bacteria. The other organelles in the cell were actually bacteria before, right?
Yep.
So we're composed of all of these bacteria that over time evolved to be together and to function as an organism, and to be differentiated with a superorganism. A superorganism is one organism, right?
Yeah.
The mold with a fruiting body, etc. Just one. But
Yeah.
us, we're actually an aggregate of all of these. So these are, Kiran, you've been with me on this journey, and you know how
Yeah.
hard it is to
It
teach
is.
allopathic medicine, about this stuff. After our lecture, for example, in the Philippines together, 5 years later, I get a call. It said, Ted, you and Kiran, when you lectured on mitochondria and gut microbiota, this is 5 years later. They are now in our society track, meaning if their societies,
Wow.
it's like 5 years later, right? It's just 5 years later, and
Lag time.
it's like that's the lag time of just teaching them and
Yeah.
getting the products like probiotics, etc., to their clinics will probably take another 5 years. So for me,
Oh
my
my
lag
God,
time,
yes.
yeah, it's about 10 years, right? You're more familiar with this than I
Oh, for sure, for sure.
am.
But the way this changes, which I've seen a lot of encouraging developments. The way this changes is really from people and consumers being empowered with this
Mhm.
kind of information, right? So listening to a program like this, getting knowledgeable on this kind of this mindset and this thinking of the body as systems biology and understanding the true root cause of dysfunction and so on, and then pushing it upon your doctors and demanding from the medical community that this is the approach that you want to see,
Mhm.
right? And we're seeing it now because what we're seeing is the biggest practices that we're seeing growing in the US at least are fully integrated practices, right? These are
Mhm.
practices that have all the integrative components, the naturopaths, chiropractors, the
Right,
nutritionists,
right.
the
Uh,
MDs, right? Yeah,
and they are the ones highly responsive to this because they wanted to do right by their
yeah,
clients or their patients, right? It's
exactly.
interesting that you note that because that's also what we're noticing in health optimization medicine and practice, right? And it's funny because we don't do the root cause of disease, that's
Yeah,
pathogenesis, right? And I ask people, do you know the root cause of health? And they don't, right? And it's like that's
right.
what we teach in health optimization medicine and practice.
Yeah,
You better come to us. It's only one, and if you get it, and you certainly get it, then
yeah,
it's a balance between anabolic and catabolic processes according to the life cycle of the organism. And
yeah.
whatever it is that you're balancing, be it gut microbiota, mitochondria, and other metabolites in the body, vitamins, minerals, and so on, you're always basically balancing the things that destroy you and the things that build you up,
Totally,
right?
yeah, yeah,
Right?
and that's so true in the microbiome as well. If you look
Yeah.
at the relationship between the intestinal epithelium, the mucosal structures, the barriers that exist in the gut, and then the microbiota that lives in the gut. The
Mhm.
the the best definition of dysbiosis, which is a term that people use to describe an imbalance of microbes in the gut, to me, the best definition that I always lecture on is really an imbalance of microbes that destroy and eat away at that lining
Right.
versus microbes that rebuild it,
Right.
right? And if there's a net destruction, then you're dismantling the system. You're creating a major source of chronic low-grade inflammation, and then that becomes kind of the foundation of processes falling apart in the body. So you're absolutely right. And the other thing I love about the health optimization approach is that to me, health is not the absence of disease, right? The absence of disease is one thing, and
Yeah.
disease is in phases and levels,
Yeah,
and
yeah,
right.
yeah. And that's
And
that's and that's by pathogenesis, right?
totally,
So
yes,
we're after salutogenesis, right?
yes,
You
that's
know,
exactly right.
yeah,
And
yeah.
so achieving health is no, it's not just preventing disease. It's not just achieving health. Health is a condition of its own that
Yes.
one has to achieve through specific means, right?
Yeah.
So Yeah.
Yeah. In fact, when I talk to my patients, I said my goal in this treatment is to get into one addiction, is to the
Yes.
addiction to the feeling of wellness. So
Yes. Yeah.
you know how it feels when you're, for example,
Oh
your gut
yeah.
microbiota are all functioning. You have
Yep.
regular bowel movements. You have well-formed stools. You have, you're actually in generally good mood. So
Yeah. Oh, absolutely. Right. When you're packed full of shit, you're definitely not going to be you're
you're
not a
also
happy camper.
you're also going to be spouting shit, right?
Yeah, exactly. Yep.
So anyway, let's get to the third one. This is a
Yeah.
short one. The gut is an enteric nervous system that is autonomous of the first brain. Thus, it is now called the second brain. In addition to this finding, the gut is now also considered to be the largest immune system of the body. What are the consequences of these findings? Kiran, when I was in medical school, the largest immune system was the bone marrow, man.
Right,
It's
right.
like, so what are the consequences of learning about these things?
Yeah, so to me, what one of the biggest consequences of this is that the immune system doesn't function on its own, right?
Mm,
It functions in the context of the microbes that exist in the body.
mm,
And one of the things I always say when I lecture on immunology is the microbes are in fact the eyes and the ears of the immune system,
mhm.
right? The if there's a positive relationship between the microbes and the immune system, then it's the microbes that are dictating to the immune system the activation thresholds, who should be attacked, who should not be attacked, what should be tolerated, right? All of that
Mhm.
important information comes from the microbes in the system, and the immune system are like the soldiers that go out there and get it done.
Right.
Right. So we've got the generals and the planners, which are the microbes, and then the soldiers and the equipment, which is the immune system.
Mhm.
And you see it all the time when the microbes are dismantled, the immune system cannot function, right?
Mhm.
And we saw this in COVID, for example, right? People who had the worst responses to COVID were people who had underlying conditions that were exemplary of dysfunctional microbiotas. So
Mhm.
people with diabetes, cardiovascular disease, obesity. There were even, I think, 4 really good published studies on mapping out people's microbiomes and then their response to the presence of COVID in their system. And what they found is that people with low diversity, low Faecalibacter prosnitzi, which is an important keystone species, low Bifidobacterium, low short chain fatty acid production, those individuals had tenfold more or more higher risk of getting hospitalized and or dying from COVID, right?
Mhm.
So, and in fact, Liam Mahoney, who is an amazing professor of immunology at University College Cork, has developed a mapping of your microbiota, and because he's been working on COVID long hauler syndrome,
Mhm.
right? He can look at an individual's microbiome and predict within 99% certainty whether what that individual's outcome to COVID was and whether or not they have long COVID
Ooh,
without ever looking at the individual,
is that test
right?
available?
Yeah, so it's a microbiome map, if you will,
Mhm.
that you can then utilize within like Biome Effects, for example, the test
Mhm.
we Microbiome Labs does. It's a mapping of certain groups of organisms and certain level of diversity. He shows that there's certain types of pathobionts. These are pathogenic or potentially pathogenic organisms that live within the microbiome, and then
Mhm.
the absence of certain keystone species. That confirmation was
Mhm.
incredibly predictive of people's outcomes with COVID and whether or not they develop long hauler syndrome. You
Wow.
know, and that's all
That's
because
very that's really cool.
yeah,
That's
it's
yeah.
mind-boggling
Yeah.
how accurate the predictions are, and the work he's publishing right now on that is really awesome because it becomes then a stepping stone to utilizing the microbiota as a predictive tool for other inflammatory dysfunctional issues, right?
Yeah.
Cardiovascular disease, Alzheimer's, dementia, the list goes on and on.
Yeah. Yeah.
There is a pattern of microbes for risk for these conditions.
I'm glad they released that because I have been forever, wanting the Grimage epigenetic test to find out when I'm gonna die, or when
Right.
when the first heart attack is. But they never released it out of I don't know what, but an abundance
Right.
of caution maybe. But it's been very accurate.
Yeah.
It's been extremely accurate in predicting all of those, and I'm glad they released something like this. And it's actually also a validation, right?
Yeah.
that it's a very important. The gut microbiota is a very important organ to pay attention to, especially because its constituents change all the time.
Yep.
They can be changed by diet. They can be changed by all of the things that it can be changed by exercise. It can be changed by
Yep.
lifestyle, right?
Just
The
geography alone,
Yeah,
right?
yeah.
Moving. Yeah.
Yeah, there. I know that in the immune system, it's only when I realized that it's the largest immune system of the body, when I realized like a lot of the kids that are born by cesarean section have a lot of allergies. That's because
Oh yeah.
they have insufficient immune activation, right?
Yep.
They don't have the gut microbiota actually. They don't swallow the gut microbiota that's needed for proper immune activation, and then
Yep.
we live such sterile lives too. Instead of parents saying to their kids, "There, go play in the mud," right?
Yep.
It's like, oh, let me alcoholize this and do that, and
Totally.
it's just like, no, we've really basically done ourselves a disservice by doing that, right?
We have,
Uh,
and there's a great example of this that I'll mention quickly. There's the Finnish allergy study.
mhm,
Finland is seeing, like a lot of Western populations, seeing a epidemic of asthma, allergies, and all that in kids, right? So
mhm,
these immune dysfunctions, where your
mhm,
immune system is lost tolerance, it's attacking all the wrong things, and so on. It's hyperactive and so on. And so they did a study where they were comparing a couple of towns in Finland that were geographically close to a couple of towns in Russia.
yeah,
And they were almost the same kind of environment. They were comparing, why does those towns in Russia have a much lower incidence rate of allergies and asthma versus the ones in Finland. And basically they understood it was all lifestyle. In Finland, they sterilize their homes. That's clean, sterile smell is the culture there. In Russia, they didn't do that. They left the windows and doors open a lot more. They had inside outside animals. So there was a lot of differences in the microbiome of the homes. And so that was a key thing. Now Finland took it one step further. They said, okay, if dirt and exposure is important, let's do a study where we take a certain number of daycares in the country and then create dirt piles and then make it obligatory where the kids in those daycares have to play in the dirt 2 or 3 times a week versus other daycare centers that don't have that. And sure enough, after a few years of follow up, they see that the ones with the dirt piles are developing kids that don't have as much allergies and asthma and all that, right? And it's just dirt. It's
yeah.
just the beauty of that, right?
Yeah, no, it's funny you mentioned Finland because I saw the happiness rankings and they're currently ranked as the happiest in the world, but they also
Yeah,
have the highest suicide rates. I said,
right.
okay, let's try to actually put this together. It's
Yeah.
like the happiest nation on earth has the highest suicide rate. So it's kind of it's like,
They
and
have
I'm sure there's a gut microbiota there in between.
Oh, totally. And also, if you think about it, right? If you're one of the people that aren't happy in Finland, you probably start to feel quite jealous that everyone else is happy and you're not. So you just end things, right? In the U.S., we probably have a lot of people in the middle that
Yeah.
are somewhat happy, somewhat not, versus like really happy people and people just want to kill themselves, right? So,
They're more extremes, right?
yeah,
They're more
exactly.
extremes.
They're more
Like
extreme.
the ones that are happy actually they upset the average because they're too
Yes,
happy. It's like putting a Bill Gates into the average income of people,
right.
right? To compute the average income of people.
Exactly.
But anyway, let's get to the fourth question. One of the biggest news, and I learned this from you, Kiran, is the biggest news is that of forbidden interkingdom communication.
Hmm.
There's evidence that outer membrane vesicles or MVs can fuse with human cells, including intestinal cells, and deliver their contents to the host cell. This can include small RNAs, which can influence the function of the host cell. What are the implications of this finding? I couldn't for the life of me do not understand why people have not placed more importance to this. This is like as Marvin Edias, who started the World Mitochondria Society, and subsequently believed that we are actually beholden to the gut microbiota. That
Right.
gut microbiota is God, is what he said. He's of course French, right?
Yeah.
And I've been attending the conferences over there, and I invited him to lecture also, and he said no. He said it's not mitochondria. It's that we're beholden to gut microbiota is God, is what he
Yeah.
said. So this thing, Kiran. So why did people not pick it up, and why aren't people doing anything more about it? Or am I
Yeah.
wrong?
No, there is some work going on with this, but it's I think you're absolutely right in that it's nowhere near the level of attention it should be garnering, because when you think about it, number one, it has dismantled one of the fundamental tools of biology, right? That this interkingdom communication is not really
Mm-hmm,
a thing.
mm-hmm,
Well, now we know it is, and not only is it true between microbes and eukaryotic cells in our case, but it's also true from plants. So there's also evidence that plants, certain types of plants, carry vesicles, carry microRNA, and all that. And in fact, some of the benefits of eating certain types of plants go beyond just the fibrous content and the polyphenols. It's the fact that these plants have epigenetic implication for
yeah.
our own our system, right? Because they're carrying these messages that can actually move to our cells and communicate with our cells. Now, when we think about the microbes, I think where this is going to start to really blow up is as we have gotten a strong understanding that the microbiota within the gut and the rest of the body impacts disease processes, impacts wellness and health processes in the body. What we really haven't figured out is how does it do it in a really significant manner, right? The
Mm.
mechanisms behind a lot of these impacts are still being elucidated. Now, I think what we'll find is that this type of interkingdom communication will become a key component of the how. And,
Yeah.
and, and that how is going to then trigger a whole body of science of understanding how do you create tissue tropic vesicles
Mm-hmm.
that can act as carriers of specific signals to take it to anywhere in the body, right? We see this. We saw this in the COVID again as another good example of this,
Mm-hmm.
because it brought up this issue of the gut lung immune axis,
Mm-hmm.
right?
Mm-hmm.
And what was interesting about that is that it turns out that certain people, when they get infected with the incoming virus into their lungs, the first things that notice the virus infecting are the microbes in the lungs, right, in the mucosal tissue of the lungs. But then the microbes in the lungs somehow communicate with the microbes in the gut, and then the microbes in the gut elicit action out of the immune system to make its way to the lungs, right? So this
Mm.
kind of interesting
Yeah.
tripart communication,
Yeah.
some of that may be done through this OMVs and microRNA and
Yeah. Yeah. Yeah.
transcription factors and all that.
Yeah, like I was gonna say, holy shit, I'm being
Yeah,
controlled by bacterial microRNAs, right? It's like I don't
yeah,
even know that my current behavior right now is just like all of the all of the bacterial RNA are
totally.
actually right. But it's very interesting. We're talking about communication here. And our audience should know that, for example, the gut bacteria and the mitochondria, they communicate. This is,
Yeah,
interbacterial communication. Bacteria communicate to each other, and we have fancy terms for them like signaling and all of
yeah,
that, but all they're really doing is communicating, right, to each other what's going on, right,
yeah,
because they are a network, right,
yeah.
they are a network in the body. So the do you think that there will be a way in the future? This is just me, blue skying here. That using bacteria as a vector to deliver, RNA, but via outer membrane vesicles. These are engineered,
Yeah.
to you know for therapeutics and doing certain communication systems in the body that would right certain imbalances.
Yeah, I think 100%, right? So if you look at the advent of mRNA vaccines, for
Yeah,
example,
yeah.
and even prior to the vaccines, of course, that's when they hit its limelight. But prior to the vaccines, they were using mRNA in cancer
Mm-hmm.
therapies and all that, right? And that's to intervene in transcriptional factors within our own genes. Now, as it turns out, bacteria are really good at that because they can create vesicles
Right.
that are tissue tropic, meaning they will secrete this vesicle in the gut, and somehow this vesicle knows how to make its way just to the brain or
Mm-hmm.
make its way just to the heart muscle or some very specific tissue. So the
Mm-hmm.
carrier they naturally create is highly tissue tropic. So what we may be able to do then in the future is understand how they engineer these
Mm-hmm.
vesicles to be tissue tropic, right? And then from that point on, it's quite easy to insert in microRNAs or other
Yeah, yeah,
transcriptional
yeah, yeah.
factors, right, and get it to those tissues. Now we've been using viruses in that way, right?
Mm-hmm.
We use
As vectors, yeah.
virus vectors because we know certain viruses go after certain tissues, so we use a virus as a vector. But I think there's complications with using viruses that way. Using bacterial OMVs, these outer membrane vesicles, are a brilliant natural way to do it. I think it could be really interesting.
Do you think we still need the bacteria then, or could we just generate like OMV-like particles to just do and target the tissues?
Yeah, I think we could probably just generate the OMV like particles if we gain enough understanding from the microbes as to how they did it, right?
Yeah.
And of course, with microbes, the beauty of it is it's so easy to do engineering of the gene. So, for example, if you find a brain tropic OMV code in a particular microbe, you can easily transplant that into another microbe and have it produce really high levels of that brain specific OMV.
Right.
Right. And then we can isolate the vesicles. Now we have these beautiful carriers that'll take whatever you put in it directly
Right.
to the brain.
Right. Right.
And that could be therapeutic compounds. It could be, transcriptional factors. It can be
Mm-hmm.
almost anything, but
Yeah.
it takes it right to the brain.
Yeah. In the formation of these OMVs, etc., there was actually something that occurred to me. I have been asked, what my position was before, and mRNA vaccines and so on. And I know that we've been studied since the 90s, right? And
Yeah.
people only learn about it now. But people like us, we monitor these kinds of things
Right.
because we know that they're going to turn out to be important. I have actually 2 points that I make. Number one is that health optimization medicine is actually about shoring up your immune system. So we take care of your gut microbiota. So the first thing that you think about when you want to shore up your immune system is to shore up your gut microbiota, so, right? Because the gut is the largest immune system in the body now, and it's in the direct interface with that. With the gut itself. And the second thing is this outer membrane vesicles. I don't even fucking know what these OMVs are putting into my system. At least I know what the mRNA vaccine is. What is pumping into my system. These OMVs, I said, the hell I know what they're putting in. Is it good for me, bad for me? I don't know, right? So it's like, okay, it's like, yeah, the devil that you know or the devil that you don't, right?
Right. Exactly.
But that occurred to me. But of course, only you would understand a joke like that. But that's my reasoning. It's like, why should I be alarmed? I don't know what OMVs are doing, right?
Yeah, totally. And they're all over our system, right? So already we just have no idea they're in there doing what they do.
Yeah.
It's going to be an interesting area of science, I think, because anytime we can target things,
Yeah.
I think it's going to become an area that people jump onto. And this is to me, OMVs are nature's just pure elegance in how brilliant nature can be, right? In doing things that we may never figure out.
Yeah, and
how it does it.
Yeah, for me, the big thing is that it just erases all the kingdom lines that are so.
Yes.
Now it proves that those are actually arbitrary, right? We put them
Totally,
there,
yeah.
and our model doesn't work anymore, right?
Yeah,
We can't say,
absolutely.
So when we're looking at, for example, when we're looking at these clinical trials, randomized clinical trials, etc., my first question was, well, did you control for the gut bacteria?
Right.
It's like
Exactly.
it suddenly throws off the entire study, right?
It shows everything, yeah.
You could just kiss the study goodbye.
Yeah.
And people don't think that way, so that's why probably a lot of people hate me. Now, the most important, the fifth one is actually the story of your life, Kiran.
Yes.
And It is the most important, I think, development in the past 20 years are the clinical developments, right?
Yeah.
So the development of clinical tests for gut microbiota, for example, stool and urine tests for bacterial and fungal metabolites and DNA, the link of gut microbiota to a wide range of health conditions, including obesity, diabetes, allergies, and autoimmune diseases, as well as lifelong health. And of course, your specialty, the development of interventions that can be used to modulate gut microbiota, such as probiotics, prebiotics, and fecal microbiota transplantation. Can you tell us your journey, not only in the science and technology, but also in this business?
Yeah, absolutely. So, I think one of the amazing things about being involved in the world of the microbiome itself is that it provides an endless array of healing opportunities and interventional opportunities that can really change people's lives, right? And it can do so for highly complicated conditions. I mean, at the end of the day, a condition that's so complicated and scary as Alzheimer's could
Mm-hmm.
have a very simple microbiome intervention to it, right?
Right,
That
right.
that could resolve that whole issue. And so being part of that and playing a role in this world of the microbiome is absolutely exciting for me. One of the things that I yeah
Let's start. But how did you get started? How did you get
yeah
interested in this?
yeah
You're witnessing of the development of the testing, because as a clinician, I can't function without tests, right?
totally
Yeah,
yeah
I know you're one of the forces they're pushing for. Like, let's we need to be able to test for this. We need to be able to do this in order to do the following studies. Can you tell us, yeah, like, where do you began and how this got you very interested and then sucked into and then sat as king on top of the world?
yeah so okay, so then my I decided to study going way back. I
Mhm.
decided to study microbiology really from a movie.
Mm,
I there was a, when I was at university freshman year of college.
mm,
There was a movie called Outbreak,
mm.
which people might remember with Morgan
Oh
Freeman,
yeah,
Dustin Hoffman, right?
yeah.
It was actually a small epidemic outbreak of a virus that came in from overseas, and this town was getting sick and all that. And
See, I
then
told you guys he's old enough. So
definitely old enough. That was way back when. The people who are listening would have to look up the oldies movies in order to find it. But what was really fascinating to me about that movie was the people that were working to kind of track down and understand this disease and come up with a
Mhm.
cure were all these people from the CDC and all. They were virologists and microbiologists, and so that was exciting to me because that was like science on steroids a little bit,
Mhm.
because it was like James Bond version of science. Because I was always a lover of science and I knew I was going to go into science, but a lot of science seems very mundane and
Yeah,
boring, and you're
yeah,
tinkering
yeah,
around in the
yeah,
lab for
yeah,
years, right?
yeah.
This brought some excitement to it. So I was like, that's what I want to study. I want to go and I want to study viruses and bacteria and things that are going to kill people. And so that's when naturally I went into the world of microbiology. Now, once I started getting into the world of research, what you start to find is that so much emphasis is put on the microbes and that make you sick, right?
Right.
And I remember I was asked during an interview once when I was still in the research world, what percentage of microbes actually are pathogenic or
Mhm.
have virulence factors? So I started doing a calculation and looking at the literature and all that. And what I discovered was that 99.99% of microbes ever discovered are either benign or beneficial, right?
Mhm.
It's basically 0.001% that are actually harmful.
Mhm.
But that 0.001% gets so much of the attention, right? And that's what everything is dedicated
Yeah,
towards,
the empty can
right?
makes the loudest noise, right?
Yeah, exactly. Yeah, in that case.
And gets the most research money. So
Totally. Always. You're going to get a grant if you're writing a grant in that world of 0.1% or 0.01%.
of
And so then
salmonella and the world of salmonella.
totally. And that's what my research at the time was actually in E. coli, infectious E. coli, and HIV.
Mhm.
So I had a virology project and a pathogenic bacterial project. Now, then I started to think that, okay, what about all the other 99.9% or 99.99%? Why, number one, what are they doing, right?
Mm-hmm.
They don't just exist with no function.
Mm-hmm.
And number two, perhaps the easiest way to control the 0.001% that makes us sick is by enhancing the 99.99% because microbes compete with microbes better than we can, right?
Mm-hmm.
And so that was already in my mindset. And then right around then, this human microbiome project kicked off.
Mm-hmm.
And this human microbiome project through the NIH kicked off with the idea of studying that other 99.99%, the forgotten ecosystem that wasn't even forgotten because we didn't even know it existed,
Yeah,
but
yeah.
the invisible ecosystem that
Mm-hmm.
potentially existed in our system. And so when I saw that, to me, that resonated as like probably the source of answers for a lot of questions that we have on how the body functions, how disease happens, how do you achieve health, and all that is because we have all of these microbes that we've completely ignored and we don't understand what they do, right?
Mm-hmm.
And so that was my impetus to get into the world of the microbiome. But then when you get into the world of the microbiome, you obviously have the drug route to go, and then or you
Mm-hmm.
have the nutritional supplement route. The drug route was a long, arduous route, right? Because
Yeah,
there's no
yeah,
models for it.
yeah.
There's almost no approved drugs for the microbiome and all that. So I'm like, that becomes a 15, 20-year slog, which may never happen,
Yeah,
right?
all we know is to carpet bomb them with antibiotics. That's it.
Exactly. Yeah, that's and everything they're going to come up with is going to somehow screw up the system more than it's going to help it, right?
Yeah.
And so then I was like, well, then you go the natural nutritional route. Then the problem with the natural nutrition route is that there's a lot of bullshit out there too,
Mm-hmm.
right?
Yeah.
Then there's the world of probiotics and prebiotics and all that. And although there are some good products, the vast majority of them have no basis behind how they're formulated, how they're
Yeah.
thought of, what strains they put in, right? Nobody's really testing the microbiome to understand what people's microbiome looks like and what may be off in their case and what should be the intervention in that case. And so for me, that became where I had to disrupt, and that became where I had the lowest hanging fruit of the opportunity to not only help people in a way, but then also disrupt some of the
Mm-hmm.
conventional norms that were driving the narrative, right? And these were narratives that I knew weren't right.
Yeah, and this is also basically a new area, right? A new
Yeah,
area where you have to invest not only your time and energy, but you also have to invest money into that
yeah,
the whole thing, right? If you are going
you
to
do.
to forge a new path, right?
Yep.
The one thing that I really appreciated of you when you lectured was that you always presented all of the research, all of your
Yeah.
research going on, other research, and that was really, really very helpful.
Yeah, and here's one way I think about probiotics, right? And this is why it always baffled me that people are willing to take probiotics that have no studies behind them and so on, and just assume certain benefit. Now, you can do that with with compounds, right?
Mm-hmm.
Vitamin C, right? If somebody's done a bunch of studies on vitamin C, and we know it has X and Y function, we know it's beneficial.
Mm-hmm.
Somebody else can come up with vitamin C as long as they can prove that it truly is vitamin C by
Mm-hmm.
structure, then you can assume the benefit is that
The
of
same,
vitamin C.
yeah.
With microbes, you can't because they're biological entities. They have
Right.
thousands of genes, right? They have all kinds of complications within them. You can't just assume that this microbe is going to be good for you because it has this name. And
Right.
so, and also, what does it do to the microbiome, right?
Right.
How does it influence your native microbes? How does it
Right.
influence your immune system?
Mm-hmm.
And to that end, here's one of the interesting things we've been discovering over the last 2 or 3 years of study in microbiome labs and then with the APC in Cork, Ireland, is the vast majority of probiotic strains that we've tested that are in the market that people use all the time are very inflammatory,
Mm.
right? They
Wow.
trigger what we call a mixed immunological profile. They trigger Th17, they trigger Th1, Th2,
Wow.
and to some degree the Treg as well. But most of them are very inflammatory because they're just microbes that the immune system just responds to,
Mm-hmm.
right?
Mm-hmm.
And so we've been dubbing this topic this question that we ask not only clinicians, but of course consumers to ask too. And the question is your probiotic silent? And that becomes really important. And the way we define a silent probiotic is one that doesn't trigger any inflammatory pathologies, but does in fact trigger IL-10 and Tregs which are anti-inflammatory
Mm-hmm.
pathologies, right?
Mm-hmm.
And so what we found, for example, the Bifidobacterium longum I spoke about earlier, that
Mm-hmm.
psychobiotic doesn't trigger any inflammatory pathologies. It only triggers anti-inflammatory. Our spores in megaspore only
Mm-hmm.
trigger anti-inflammatory. And so when you think about that and you go, okay, I'm a clinician. I have a patient that's dealing with an inflammatory condition, which is what
Mm-hmm.
most long-term or chronic issues are. The last thing I want to do is give them a probiotic that's inflammatory,
Mm-hmm.
and now clinicians, when I talk about this, clinicians go, that makes sense. This is why my MCAS patients, right, my mast cell activation patients, or my
Mm-hmm.
histamine intolerant patients, this is why probiotics make things worse for them,
Yeah.
right? More often than not, it's because they are in a deep inflammatory issue, and you're giving them a pro-inflammatory compound,
Mm-hmm.
right? And so that to me just underscores why it's so important to focus on research, to know what these biological entities are doing, what functions they have, how they impact the immune system, how they impact your microbiota, and so
Mm-hmm.
on. And 99.9% of companies aren't doing that.
Is it still in your opinion? There are still live probiotic vendors out there, etc. Do those have different effects than, for example, a spore probiotic? And is it worth investing in them for our patients, our clients, for example?
Yeah, there are some that are really, really beneficial, right? There are
Yeah.
some strains, for example, Lactobacillus acidophilus DDS1. There are
Mm-hmm.
tons of studies showing that the strain, both in live form and actually attenuated form, dead form, both forms actually have functionality in the body and do attenuate certain processes. There are numerous strains, 35624. Bifidobacterium longum is another one. So they are, and I think the key to it is that understanding which specific strain has the functionality and what the functionality is, and then using that for that condition, right?
Mm-hmm.
To me, the idea of these kitchen sink probiotics, 20, 30 different strains at 100 billion, 50 billion, all of that kind of a dosing, that is where we start to falter because we have too many things in there. We don't know what they all do. We don't know how they all interact with each other, and we don't know how they all impact the microbiome. One
Mm-hmm.
example of that I'll give you, that Bifidolongum strain is another example, right? We know that that strain has very unique effects on the brain, brainwave function, like we talked about, activating the parasympathetic system, reducing inflammation,
Mm-hmm.
all of these beautiful things it does. If you mix that strain with another Bifidobacterium, it knocks out its effect,
Oh.
right? And so the microbes compete with each other
Yeah, they do
and they
compete. Yeah.
create competitive forces, right? And
Yeah.
so we've done studies now where we mix this strain that does all of these amazing things. We've got 10 or 11 publications on what this strain does. You mix it with one other probiotic bacteria, the effect is gone,
Out. Yeah.
right? And you
Yeah.
would only know that if you did the studies on it.
Yeah, that's why I made a quip earlier on the AI library model, language library model that I was large language
Yeah.
library model so working on. When I asked it what are the three C's of evolution, it's like
Yeah. That's
it's
right.
competition, cooperation, or compromise, and
Right.
apparently with your experiments, it's competition, right?
Yeah,
So
it is. Yeah.
it's gonna I'm down. Can you tell us what are the most important tests for you that are deliver significant results right now, for example, from the gut metabolites to the gut bacteria composition, gut bacteria metabolites to gut bacteria composition, and
Yeah.
all of these other things that you look at clinically, when you
Yeah.
are doing a clinical trial on a patient, for example, on patients, for example, or on subjects. What are the significant ones, and how were they developed?
Yeah. So to me, one of the most important is having a clear way of looking at the metagenomic analysis of the gut microbiota, right?
Mm,
Now, it becomes really hard at the species level to really distinguish what's affecting what.
mm,
However, when you go up to even the genus level or even beyond the genus level, things like the phylum level, you start to see patterns emerge that can be really, really profound and important to understanding not only how a condition is impacted by the microbiota, but then how impacting the microbiota can alleviate the condition,
Mm-hmm.
right? And we did this in a good example of that is in acne.
Mm-hmm.
We've done and published the biggest probiotic acne study ever, and we developed a probiotic called Serene Skin that reduces acne lesions by 70% in 12 weeks, right? As
Wow.
good as any antibiotic
Yeah.
that's used for acne, without, of course, all the side effects of the antibiotics. Now, how did we come about that? We came about the discovery of this largely through understanding what the microbiome looks like in the case of acne. So
Mm-hmm.
we took a bunch of acne subjects and we started doing full whole genome sequencing of their gut microbiome through stool testing. Now, the stool tests are have limitations, so we
Mm-hmm.
have to take them for what they're good at doing,
Mm-hmm.
and they're really good at looking at certain patterns of groups of microbes, right?
Right.
And so what we started seeing in the test that we developed, and we developed a test using whole genome sequencing, shotgun whole genome sequencing versus 16S. 16S, of course, looks at certain variable regions of microbes.
Mm-hmm.
It's low accuracy when you're trying to get to the genus or the species level,
Mm-hmm.
and the probability of finding the right answer in that is much lower. So we do shotgun whole genome sequencing, which
Okay.
is looking at the whole genome of the
Yeah.
microbe from end to end. And so what we did is we started looking with acne patients, the whole genome. We came to see that most people with acne that we tested had very low Rhamnococcus levels and then had very low levels of acetate production, which is one of
Mm-hmm.
the three main short chain fatty acids, right? So
Mm-hmm.
they might have okay butyrate production, but then
Mm-hmm.
converting that to acetate, they tend to be deficient in. And that started to make sense because number one, acetate goes outside of the gut. It's a short chain fatty acid that functions largely outside of the gut. You find it in other tissues in the body, and you find it in the mucosal layer of the skin underneath. You
Mm-hmm.
find it in the sebaceous gland and so on, reducing inflammation and helping repair tissue and so on. So we said, okay, that makes sense. So if acne patients had low Rhamnococcus, low acetate production, what if we gave them a probiotic that actually had that could enhance the growth of Rhamnococcus and that could enhance the production of acetate? So we
Mm-hmm.
created a spore-based probiotic that did that.
Mm-hmm.
And sure enough, in the follow-up studies that we did, we saw not only were we increasing Rhamnococcus, we were increasing acetate production. That also resulted in a significant reduction in acne,
Mm-hmm.
right? So what was great about that process is, of course, it's wonderful that we came up with a safe, effective way of
Mm-hmm.
improving acne without being on antibiotics for 140 days, right? The great part about that is the clues for how you develop the intervention came from the microbiome, came
Mm-hmm.
from understanding what the microbiome of acne subjects looks like,
Mm-hmm.
right? And so for us, we've been doing that now in other areas as well. We've looked at what does the microbiota of rheumatoid patients look like, right? What does the microbiota of obese individuals look like?
Mm-hmm.
What are the characteristics? And then if we modulate those characteristics, can we change the outcome for those patients?
Mm-hmm.
And the beauty of that to us is that if we can maintain that modulation, maintain the positive change, then we should be creating a long-term impact on those patients rather than maybe a short-term intervention. In the case of weight, this can become really important
Yeah.
because how can you give someone an opportunity for long-term weight solutions versus all the kind of the things that people do that are yo-yos and so on, right?
Mm-hmm.
So that's to us the gut microbiome testing became a really important part. And so for every clinical trial that we do, even if it's not directed by the microbiome, we add in microbiome analysis because we want to understand how the intervention impacts the microbiome, and then does that impact have the clinical effect as well,
Mm-hmm.
right? As we start collating all of that data, I think as a scientific and medical community, we'll just become much smarter at what
Yeah.
we do.
I have two questions, Kiran. One is on, currently looking at addiction patients, say alcohol
Yeah.
addiction patients. Do you think that microbiota we could essentially sequence or do an analysis of what is driving in addition to context and other such learned behaviors, right? Is Do you think we can help alleviate these kinds of addictions,
Yeah,
because it's very difficult to treat, as you know.
it is. Yeah.
Um,
I absolutely think so. And in fact, we were working on a project with a recovery group that manages meth patients, right? So this was
mm,
we weren't looking at meth. We weren't looking at alcohol. We were looking at meth in this case. But I think addiction, there's a lot of underlying similarities, no matter what the substances
mm,
you're addicted to, right? I think the tendency to have addiction in itself is likely a dopamine issue, and that dopamine issue likely comes from a dysfunctional microbiome
mm,
that's not producing enough dopamine, and or a microbiota that's negating the function of dopamine in the brain. Because one of the things we know is elevated levels of endotoxin, for example, in circulation
mm.
from a dysfunctional microbiota interferes with dopamine receptor binding,
Yeah, or
right?
from leaky gut, right?
Leaky gut,
Yeah,
exactly. So
yeah.
leaky gut itself could screw up your body's ability to utilize dopamine,
Right,
right? And then at the same time, a leaky gut is likely not making enough dopamine. So
right.
now you become addicted to dopaminergic behaviors,
Mm,
whether it's food or alcohol or substances that stoke dopamine to make you feel normal, right?
mm,
And unless you cure that deficiency, then you're always going to have a tendency to go back to addictive behaviors, right?
mm,
Even if you can modulate the chemical dependency that people
mm,
end up getting with alcohol, with heroin and so on. You can modulate the chemical dependency, but that underlying driver, the motivation for these
mm,
individuals to
mm,
seek
mm.
dopaminergic substances will still be there,
Yeah,
right? And I think that's part of the relapse.
yeah, that's actually, I'm glad you said that, because if especially for alcohol addiction, this is a continuing, this is a
Yeah,
daily thing that you
it
have
is.
to. And I have had patients that basically it's a struggle for them daily, and it's
Hmm.
always a reassurance, right, that
Yep,
this is a disease. It's not, an
totally.
addiction is a disease. It's not them that's a failing, but it's a disease as a hold on them. And so if there's something like, aside from lifestyle and so on, there's something that we could give every day. I was thinking that maybe it's probably going to inform of some gut microbiota that can actually do this
Yep.
brain neurotransmitter modulation, right?
Yeah, and it may be a form of fecal microbiota transplantation, right?
Mm,
Because
mm,
there are certainly people that are extremely resilient to against addictive behaviors, and then
mm,
there are people that are absolutely prone to addictive
mm,
behaviors. Right now, I have friends like that. Friends on both sides of the spectrum. I have
mm,
friends that can, do things that normally would be addictive to other people, and have no desire to be addicted to it at all. And I have friends who can barely walk by, a marijuana dispensary without obsessing about all the things that they need to buy, and sitting there and smoking and eating gummies all night long, right? And so there's like spectrums of this.
Yeah, the reason, right, that came to mind to me as a possible solution is actually when they used fecal microbiota transplant uh, for C. diff, right? For
Yeah.
proceeding with C. diff in an adult, and then afterwards, the recipient actually got cured of the C. diff, right, and started to get obese.
Yes. Yeah.
Right,
Exactly.
remember that case,
Yep. I
right?
do.
It started to get obese, and it's because actually the microbiota was obesogenic in
Yep.
composition, right? And
It
then
is.
I think I don't know, there was a lawsuit or something like that, right, that followed. And I said, well, if it's able to make you fat, right,
Right.
is it able to make you stop drinking alcohol or whatever it is that you're addicted to,
You're addicted
right?
to. Yeah,
Yeah.
absolutely. I think so. And there have been individual cases on things even more profound. Like there was a cerebral palsy case
Mm,
where the palsy was alleviated by doing a fecal microbiota transplant. There are people with really complex neurological disorders.
mm,
So lots of different conditions that have now been demonstrated to be modulated by changing the microbiota. And
mm,
so, with that in mind, I think tendencies like addictive behaviors and all that are absolutely within the spectrum of things that we could probably intervene on for the right microbiome modulation.
Kiran, can you touch on briefly on if you know of any studies being done for autoimmune diseases? Since for the life of me, one of the more difficult cases to treat, of course, is chronic fatigue syndrome, or
Yeah,
now called myalgic encephalitis, right?
yeah.
And one of the things that gives an immediate relief, actually, to my patients is when I clean up the gut. I
Yeah.
you seal up the leaky gut, you clean it up, you basically make sure that they have a more diverse gut microbiota. And in relation to that, Kiran, can you explain also, have we already found like the core microbiota that's common to all human? Is it true that
Mm,
60% is the same, or what has happened to that project? Has it ever been resurrected? That we will
right.
ever find the common microorganisms? So these are the kinds of things that I don't know, but you're the expert in this.
Yeah, no, no. This is all really exciting areas. So in the world of autoimmune, we completed a study on rheumatoid arthritis
Mm-hmm.
as a really strong model of a complex autoimmune condition. And in that study, I only have the preliminary data yet, but so far what we're seeing is a really powerful um, ability to intervene in the disease process, where not only are we reducing pain, reducing inflammation as measured through pain scoring and inflammatory markers, but we also ended up doing scans of the joints
Mm-hmm.
to actually visualize the reduction in swelling in the joints. And then, of course, we have mobility measurements and all that. So we're improving people's mobility. And we were doing it all with the spore-based probiotics in this case, because one of our hypotheses was that a low production of short chain fatty acids and a leaky gut was a significant driver
Mm-hmm.
of the risk factors underlying rheumatoid arthritis. And then the other component, which I think is probably one of the most important thing, is the dysfunction of the T reg system, the T
Mm-hmm.
regulatory cells, right? Because ultimately, it's these T regulatory cells that are supposed to be intervening and eliminating or dampening all of these unfavorable immune responses, right?
Mm-hmm.
These immune responses that go haywire, whether it's something as simple as allergies, asthma, or something as severe as a rheumatoid arthritis, or severe eczema, psoriasis. All of these conditions are a haywire immune system that need to be regulated,
Right.
and the regulatory part of the immune system, called the T reg system, is
Right.
highly dependent on a diverse microbiota to enhance its expression.
Mm-hmm.
And then the T reg cells are very dependent on short chain fatty acids for
Mm-hmm.
fuel to
Yeah.
function, right?
Right. Right.
So without high diversity in the microbiome, without high production of short chain fatty acids, and with leaky gut, if you have leaky gut and you have low levels on the other two, you're absolutely set up for autoimmune risk. And
Mm-hmm.
to me, that's going to be one of the critical ways in which we intervene with autoimmune conditions, is by increasing diversity, increasing short chain fatty acids, and reducing endotoxemia or leaky gut and the resulting inflammation. Another area that's been really interesting is neuroinflammatory conditions,
Oh, yes.
right? When we've been
Mm-hmm.
studying our psychobiotics in
Mm-hmm.
the neuroinflammatory area, because as it turns out, with a lot of neuroinflammatory conditions, there's a tryptophan dysregulation that's going
Mm-hmm.
on, right? Where tryptophan, instead of being converted into melatonin and serotonin, is being converted into quinolinic acid,
Quinolinic acid. Yes.
right? And quinolinic acid is very inflammatory, very neurotoxic. This seems to be the case in issues like fibromyalgia and people with chronic pain. This is also the underlying mechanism in COVID long hauler syndrome,
Mm.
which is really thought of as kind of autoimmune like condition. So we've been using the psychobiotic, the 1714 strain, because as it turns out, that carbohydrate structure that it possesses shifts tryptophan metabolism back to
Mm.
serotonin and melatonin, averting quinolinic acid. right? So if you can do that foundationally,
Yeah.
that I think is going to be a huge improvement in people with all these neurological inflammatory conditions. The other interesting thing is Ted Dinan, who's this integrative psychiatrist who played a big role in discovering this psychobiotic. And in fact, he's the guy that coined the term psychobiotic in a book he wrote called The Psychobiotic Revolution. One of the things he's discovered recently in a study is that it's likely over 90% of the tryptophan that we use in our body comes from microbiome synthesis, not from diet. We can't assimilate tryptophan well
Right,
from the
right.
diet, right? And not from other sources. So if we don't have enough diversity in the microbiome to make tryptophan, which is a critical thing for neuroprotection, then we're going to be at risk of neurodegenerative conditions. And then the tryptophan that's being made, if that's being shuttled into quinolinic acid, then we're driving a neurotoxic condition, right? So this is where the microbiota plays such an important role in all these conditions.
Is the product already out?
Yeah, that product's called Zen Biome.
Oh, okay. I have
It's
I have
out
a
in the
couple of
market.
bottles here.
Yeah.
I haven't checked the label though. Does it contain vitamin B6? Hey.
Yes, it does. Actually, it does.
I haven't checked the label. But
Yep.
for those like Kiran, we know that the cofactor that drives the vitamin that drives these reactions of
Yep.
tryptophan, uh, you know, to hydroxytryptophan and so on is actually driven by vitamin B6.
Yep. So we put it in there. Absolutely.
Yeah,
Yeah.
yeah.
And then your question on the kind of the core biome, right?
Yes, yes, please.
So there is more work going on this. I think where it started to deviate was a number of researchers in this space started looking at the microbiota of indigenous tribes and trying to understand how the human microbiome has evolved over the last, let's say 10,000 years, right? From hunting, gathering, foraging to where we are now. What are some missing microbes that were core to the biome then that we don't longer have now? And so that has actually provided some really, really interesting insights. I think one of the things that is absolutely certain is that we are losing diversity in the microbiome at an alarming rate. Right. And we talked about earlier how our microbiome houses the vast majority of functionality that we have as a species. And it does so through the genetics, and it does so through peptides and everything else that it produces that we can't make ourselves. Now, as we start to lose large portions of that ecosystem, we lose functionality. And the analogy I give to people is imagine we told somebody that based on your behaviors today, the next generation of kids will be born without a spleen, right? And the generation after that would be born without a spleen and only one kidney, right? Those are organ systems, and everyone appreciates the importance of organ systems. And being born without an organ means you're born with a significant deficit. But now we know that the microbiome is an organ, right? And so if we are not harboring and protecting the ecosystem within ourselves and passing on an adequate ecosystem to the next generation, we are effectively causing the further generations to be born with a severe handicap, with missing components of this critical organism, right? Or sorry, organ system.
Yeah.
So we know that diversity is king. And alpha diversity. So that is the number of viable species and their uniformity within the gut microbiota. So that reigns true across the board. But we still do know that at the species level, people are vastly different, right? But what is being realized is that even though people are vastly different at the species level, functionality seems to be conserved, right? Like you and I could have maybe 30% similarity in our microbiotas at the species level. And yet, if we're both healthy, we have 98% functionality similarities, right? Meaning there's so much functional redundancy within the microbiome. The microbes producing short chain fatty acids for you may be different than the microbes producing short chain fatty acids for me, but yet we both have short chain fatty acid production, right? So that's the part that's becoming really interesting to understand, is that the microbes themselves may be different from people to people, but the functionality has to be conserved. And if you're missing functionality, that's when you start to see disease process occur. So if you have a group of microbes that produce short chain fatty acids for you, I have a different group of microbes that produce short chain fatty acids for me,
Mm-hmm.
we're both fine. But
Yeah.
if a third person has very few microbes that produce short chain fatty acids, that's going to be the person that's in trouble,
Yeah.
right? They don't have to have your microbes, they don't have to have my
Yeah.
microbes,
Yeah.
but they have to have some
The
microbes
microbes
doing
that are
it.
producing. Yeah.
Yeah.
Yeah. It's
And so
evolutionarily built, right? Redundant systems.
it is.
Yeah.
The
Yeah.
redundant systems are
Yeah.
critical, right? And we see this in other animals as well. There was a great study done on in the veterinary space on chickens, where they showed that from the day the chicken is born
Mm.
to the day it's fully grown. And in the case of chickens, it's only something like 68 days. In that short period of time, the microbiome of the chicken changes drastically when you
Mm-hmm.
look at the species level, right?
Mm-hmm.
Yet the functionalities remain the same,
Mm-hmm.
right? So certain microbes die off, others take over that do the same thing, and so on. So now, when you think about it that way, then to me, a microbiome analysis and to understand the health of your microbiome, we have to look at it from the perspective of functionality and functional groups,
Mm-hmm.
right? And not what microbe you may or may not have as individual microbes, right?
Mm-hmm.
And so when we did design, for example, the biome effects test, not only did we go with whole genome sequencing and have the most accurate genetic sequencing technology, but we designed the tests around. So I almost dropped my iPad here. We designed the test around identifying functional groups of microbes. Right? We looked at your short chain fatty acid producing group versus my short chain fatty
Mm-hmm.
acid group versus another version of short chain fatty acid groups, and we give you the relative abundance of that group of short chain fatty acid producers in the context of the whole microbiota, and we look at that as a measure of health. So if you have 13% of your microbiota, let's say, are short chain fatty acid producing bacteria, and that proportion is associated with health, it doesn't matter necessarily what microbes those are, as long as you have them at the right level, right?
Okay.
And if you have 6%, that means you're highly deficient in short chain fatty acid
Mm-hmm.
producing microbes, right? We need to do things to enhance that functionality. If you have 30%, maybe you're a little too high, and maybe we need to bring those down, right? And maybe it's high because now you have a lack of lactic acid producing microbes, right?
Mm-hmm.
Which would be the precursors
But
to
we do
short
know
chain
what
fatty acids.
we do know already what those functional groupings are.
We
Or
do,
are we,
yes.
we do. Okay.
We do, and then we're getting better at it almost every month
Mm-hmm.
because there's lots of great research coming out identifying microbes, doing gene blasts of the microbes, identifying all the proteins and everything that the microbes can make, and then that data goes into a bank
Mm-hmm.
that we reference against, right?
Uh-huh.
And so if we find microbe A in your system, and we're trying to figure out, well, what functionality does microbe A have, we can then reference that against a genome banking of that microbe, and we go, this microbe produces vitamins, it produces this enzyme, it produces short chain fatty acids. Great. So now we know that that microbe adds to your functionality of those components, right?
Mm-hmm.
So those discoveries are getting better and better every single day, and it's really about staying up with the data sets.
Mm-hmm.
As the data sets improve. We looked at the data set even a year ago, it would look wholly inadequate compared to
Yeah.
the data
Yeah.
set
Yeah.
today.
Yeah. You said something earlier about foundational organisms, right?
Yeah.
What do you mean by foundational organisms? And are they do they belong to the healthy set, or do they belong to the 0.01%, which if you actually
Ah,
have them, you're going to develop this? So what exactly do we mean by foundational organisms?
yeah. So that's a really important one. So these are what we refer to as keystone species,
Mm-hmm.
and like any other keystone species in any other ecosystem, they become responsible for a couple of key things. Number one is they become responsible for maintaining the stability of the ecosystem, right?
Mm-hmm.
So just like in any ecosystem, if you remove an apex predator, for example, much of the ecosystem will start to fall apart
Right.
because everything kind of flows up towards that
Right.
apex predator, right? These species aren't predators, but that's just an example. These keystone species have been shown to be inversely correlated with lots of disease conditions. So, for example, Faecalibacter prosnitzi is a great one.
Mm-hmm.
Everything under, Crohn's, colitis, microcolitis, colorectal cancer. If you have healthy levels of Faecalibacter prosnitzi, then you tend to be protected against all those conditions
Mm.
because F. prosnitzi plays a very important role not only in producing short chain fatty acids, stimulating the production of mucosal layer, but also maintaining diversity within the gut microbiota. It's a balancing force
Mm-hmm.
within the gut microbiota. So there's about 11 or 12 microbes that have been identified as kind of keystone organisms where their presence is automatically associated with health,
Is
right?
Akkermansia one of them or no?
Acromantia is one of them.
Muciniphila?
Acromantia and muciniphila, yep.
Yeah.
Bifidobacterium longum is another
Yeah.
one. Bifidobacterium animalis is another
Mm-hmm.
one. There's a Rubinococcus species that is as well. So there's a few of them where individuals that are missing these keystone species tend to have elevated risk of certain types of chronic conditions.
Is it actually bad, Kiran, to put all of these known foundational organisms, microbes, into a single, spore supplement?
Like a supplement, the biggest problem with most of the keystone species in terms of supplementation
Mm-hmm.
is that most of them are what we call obligate anaerobes, right? And
Oh,
so they cannot
yeah.
function in oxygen, right? They
I
die
see.
within seconds of being exposed to
I
oxygen.
see.
And many of them, like Akkermansia, for example, is not known to exist anywhere but the human gut,
Mm-hmm.
right? And so
Oh,
it
yeah.
plays this really unique role of just existing only in the human gut. So
Mm-hmm.
the moment you take them out to try to cultivate them and grow them and all that, they automatically die.
Mm-hmm.
And they have very little capability of actually surviving through the gastric system and engrafting into the gut microbiota. So when we've worked with a number of them, we've worked with Akkermansia, we worked with Butyriccoccus polycecum, we've worked with a few of these obligate anaerobes, and what you come to find is that they're really, really finicky.
Mm.
They're very unstable, and they're not at all designed to be outside of the body. So really, then we have to look at, well, what can enhance the growth
Mm-hmm.
of these organisms endogenously? And I actually believe, I don't have the data to support this yet. I have some small tidbits of data, but I actually believe that the vast majority of people have most of these keystone species, even if it's at such a low level where they're not functioning yet,
Mm.
right?
Are they hidden in the crypts of the intestines or something?
Exactly, yeah. They're in there, they're at a tiny, tiny percentage,
Yeah.
right? So you look, for example, at a healthy individual can have upwards of 5% of their total microbiome as Akkermansia,
Yeah.
right? Now, if you can have Akkermansia and have it as 0.1%, it's still there,
Yeah.
but it's not doing anything. Its
Yeah.
numbers are just too low. And so then the question becomes, how do you enhance its growth?
Mm-hmm.
Well, as it turns out, there's nutritional ways, there's lifestyle. For Akkermansia, for example, fasting, we know, will increase the growth of Akkermansia, right?
Mm-hmm.
We know that polyphenols increase the growth of Akkermansia because Akkermansia likes to consume polyphenols. We know that certain oligosaccharides will enhance the growth of polyphenols. So that's where the modulatory capabilities come in, where we can intervene with lifestyle, behavior, nutrition to enhance the growth of these critical keystone species.
And that's why there are dietary studies, right? From various
Yep,
cultures, you study the various diets because the diets have an immediate effect
absolutely.
on the ecosystem, right? And
That's absolutely
from
right.
what you're saying, I'm a biology major and I took ecology, and one of the things that's actually interesting here, working on having a background in networks in ecosystems, and then background on neural networks, is that we take a look at the architecture. It's basically
Yep,
ensembles of networks, right? These are
yep,
ensembles of networks that are communicating with each other. So within an ecosystem, there are micro networks in there that are doing their part, are doing their function, right?
that's
So
exactly right. Yeah.
yeah, and that's why it's such a, as you said, it's a difficult area to be able to make a dent in. But Kiran, you made a fantastic dent.
Thank you. Thank you
you,
so
you
much.
you made a fantastic dent. You've devoted so many years in your of your life to this. Have built a successful company, and now I heard that you're now out of the corporate life. So what's next for Kiran?
Yeah, and it feels really quite amazing. It's been a heavy lift off the shoulders to be able to be free of that and feel comfortable that you've put the right leadership place in place to take that on, into the horizon. But what's next is really resolving other problems where I think some serious intervention is needed, disruption is
Like world
needed.
peace, is that world peace?
Exactly. Yes. The whole Palestine Israel issue still needs to be solved, right? I think there's a hole in the ozone somewhere that needs to be fixed.
Yeah,
so
but
so
I heard
that,
you've started now another couple companies, or
yeah, we're in early stages. A couple of them will be launched this year. Couple of the areas. One of them is in the world of hormones.
Mhm.
Hormone imbalance is a major
Ooh,
issue, as you know, right?
one of my favorite
Um,
things.
Yeah,
Uh,
exactly, and it's
that I
it's an
do.
area. Yep, it's a world that you're an absolute expert in, and there's a couple of really good tests right now for hormones to really look at it in depth. One of them is called the Dutch test, and the
Mm.
Dutch test is a very comprehensive urine metabolite test.
Hmm.
The problem with it is that most practitioners cannot make heads or tails of it. It's too
Yeah, and,
complex,
and it
right?
also cannot detect protein-based hormones,
Right. Yep, exactly. And so you need to be able to add in blood, you need to be able to add in
right?
saliva and so on. So what we've done is we've created an AI that can read that all 17 pages of the Dutch test and
Mm-hmm.
then incorporate blood and
Mm-hmm.
saliva as well as needed,
Mm-hmm.
and then really provide a true picture of what the pattern dysfunction is in this individual, right?
Oh, cool. I'll train your LLM for you and make sure it fights with me.
Well, I'm sure it will. I figured you would have a feisty LLM anyway, so that's fitting. Because what we want to do is really get to the understanding root cause of the pathologies around hormone disruption
Mhm.
and dysfunction, whether it's earlier in people's lives where they're being disrupted by all these, phytochemicals and xenochemicals
Yeah,
and all that, right? Xenoestrogens and all that, or it's
yeah,
people who are dealing with a stage of the biology where
yeah,
hormone disruption is an
yeah,
issue, right?
yeah.
Yeah,
ADCs, endocrine disruptors, right?
exactly.
Uh,
All the endocrine disruptors we're
chemicals.
surrounded
Yeah,
by.
yeah.
Uh, but here's an interesting thing we found, and now that we've been going through this and we've been developing the AI and coding it and all that, we found that in women alone,
Mm.
there are at least 350 different pattern disruptions in hormones in the perimenopause and menopause stage,
Wow.
right? 350 different types
Mhm.
of disruptions that you can find.
Yeah,
In men, it's about
is the
40.
husband or sexual partner one of
Yeah,
those? Is
exactly. Well, as it turns out, the husband has about 40. So it's, somehow he's imparting much of the complication to the woman, unfortunately. But it's just the science around it is highly complex, and I think like you've identified, for certain things you're going to need AI to really
Yeah,
understand this.
yeah.
And my big picture goal is that there's lots of complicated chemistries and tests. We're going to need an AI to help really understand each one of them. And then we'll need a central AI to take inputs from all of those to look at all of the overlying patterns, right? To get a complete picture of the individual. And that's just because, and that's the limitations of the human brain, right? No, not one person can be an expert in everything. And
Yeah,
so,
yeah.
and this is why we have silos of medicine, right? You deep dive into one
Hmm.
area,
Hmm.
you can't be an expert in everything. And so we can create AI experts in everything, and we can create a central AI that actually takes all the inputs
Hmm.
and understands the true complete pattern of the individual. So that becomes a big passion project for me to try to bring this kind of thing to market.
That's because you're getting old,
And
and
getting old, exactly.
and andropause is on the horizon.
We've got to do anything we can do to intervene here.
Well, you're talking to the right person.
Yep.
Anyway, I know I lecture on the fact. My interest in this, Kiran, is actually more I lecture on the fact that we are, the hormones actually drop, as
Yeah.
we age, as a sort of like a normal process of aging. And over the years, it's all finding out about why these drop and so on and so forth, and how could be supported by nutrient cofactors and other,
Yep.
botanicals, gut microbiota, optimization and actually putting in the hormones themselves. But in the end, it's actually that's finding out why this, the enzymatic processes declined with age and so on and so forth.
Yeah.
So there's a lot of that there, and when I present my slides, it's like it's all of these things just going down, right? Your
Yep.
your growth hormone, your thyroid hormone,
Yep.
your insulin levels,
Testosterone,
testosterone,
everything. Yeah.
everything just going down. And I tell them, no, this is not disease, dear. This is just normal getting old.
Yep. This
So
is
so
old age.
yeah, what's your other project? So you're doing hormones, and what's the
Yep,
other one?
dealing with the hormones, and we have a couple others. One is I want to bring a new level of traceability and technology to phytoceuticals, so plant-based
Mm-hmm.
active compounds, right? And what we want to do is basically utilize things like blockchain technology to provide really good traceability as to
Mm-hmm.
where that plant came from.
Oh yeah,
You
yeah,
know,
yeah.
yeah, the exact GPS coordinates, for example,
Right,
on the farm,
right.
who the farmer is, understanding what they use in the soil, having good data on the health of the soil during the production of the plant, right? Including things like contaminants and so
Yeah,
on.
that is so important.
Yeah,
I don't know whether I told you the story where I read this article on Tonkat Ali. You recall
yep,
Malongifolia that's
yep,
supposed to increase testosterone levels, etc. The ones that they did in the study was actually growing at the foot of the mountain.
yeah,
I think at the foot of the mountain. I may have this in reverse, but the ones that they harvested were at the middle of the mountain.
mm,
And so the studies were positive. And then when they actually turned it into a product, and it was tested, it was no
yeah,
longer active, right? So the place where it's grown, the amount of
yep,
sunlight it has, that's why this project of yours of actually identifying where it's growing, what the soil conditions are like, what the toxins are, and so on. They're really very important. And this is the way he to my audience here, this is the way Kiran makes me fall in love with him, because he's pushing the right buttons. Because I love medicinal plants, and
yes,
that
absolutely.
has always, we're I think we're the only two people in the world who call it phytoceuticals all the time.
I mean, it's to me, there's no better way to describe it. I mean,
Yeah,
they're more powerful than any pharmaceutical,
yeah.
and they're natural. Your body knows what to do with these things, right? When it encounters it, and so I want to bring not only a degree of traceability
Mm,
and background on this to understand where the source of it is and what it looks like and so on, but then we're also looking at can we enhance extraction processes and all that
mm,
to really isolate the true active ingredients, right? And with
mm.
plants, in many cases, it's a matrix of things that's
Right.
important, right? Even though there may be one ingredient that gets a lot of the credit,
Mm.
like in Boswellia, it may be AKBA, but then
Yeah,
there's a matrix that comes along with
yeah,
it
yeah.
that's important. So being able to identify these components of it and then ensuring that we're doing the right types of extraction process so that we can maximize the benefit of these phytoceuticals. And I think that's very needed because small molecular synthetic chemistry is going to go on, but
Yes,
it also has limitations, right? Because there's certain economic limitations around patentability of these
yes,
compounds,
yes. I
right?
agree. I
And
agree.
if the compounds aren't patentable, companies aren't focusing on it. And so, what happens to all the diseases where these companies aren't seeing the patentability of a compound to intervene? We already have a whole category of medicine called orphan drug
Right,
issues there,
right,
right?
right.
Where pharma companies are just like, well, it's not even worth our time investigating those diseases because the economics don't make sense for what they have to do. But plants likely have a solution for almost all of these things,
Yes, I agree.
right?
I
If
agree.
it's done correctly, right?
Yeah.
And so we want to set a benchmark and bring a model to phytoceuticals that hopefully can be replicated then by other companies in the space
Yeah,
too.
because as you know, I am the chief formulator for prescriptions products, right?
Yeah,
And actually, you have several products, but the one that's right now that's pivoting as an anti-anxiousness for formula
yeah.
has kava in it, right? And kava
Oh yes.
lactones, and it's really hard because as someone trained in pharmacology, I want to know exactly how many milligrams of,
Yep.
cavein is actually going into the troche that I'm formulating, right? When
Yep.
you say 65% cavalactones, what exactly does that mean,
What does that
right?
mean?
What
Yeah.
does that mean?
Hmm.
So these are the kinds of things that I'm grappling with, and it's actually funny that we're in the world of phytoceuticals. We're coming up with a sleep product, and I've been working with Magnolia officinalis, right?
Yeah.
And honocayol is the active substance in there, and it is so fucking sticky, man. So
Yes. Oh
so
my God, it
so,
is.
so you formulate with it, and it just sticks to your hand. It sticks to
Yep.
everything else. It's just this thing. But and you begin to realize that these are the very things that you need to be able to dose properly, ascertain the purity.
Yep.
Know where it was extracted. Uh, spend a lot of money spending for toxicologic studies, right? To see or to see what are the components in there. And this is your project, so
Yes.
this is really cool.
Yeah,
So
yeah. This is another one that we're dedicating a lot of time to, and and I think again, this will be disruptive to the world of phytoceuticals, and as it needs to be, and then we want to create a benchmark of how you do this.
yeah,
How you do this properly. How do you use technology to understand the traceability? How do you understand extraction processes better? Because the sticky component, for example, the active ingredient may not need to be sticky,
yeah,
right? It's
yeah,
bound in these resins, and
yeah,
so
yeah,
is there another way of extracting the key component without having all these
yeah,
resins
yeah.
that makes it difficult to work with? So we want to improve all of this, and I think that's going to be a beneficial service, I think, to the industry, and then it'll allow magicians like you to do your thing, in a easier way, to do your formulation miracles and make
Well,
people happier.
you're the master magician. I'm just apprentice, an old apprentice. And Kiran, before I ask you the final question
Yeah.
here, I would first like to thank you. It was
Thank you.
really incredible to have met you and interacted with you all these years, and it's been a wonderful ride.
Yeah.
Uh, and we're still here, right? We're
Yep.
still spreading the gospel about all of these things that only now we know matter to us. Right?
Yep.
It's like
Exactly.
what you don't know will hurt you. So
Yes.
the second thing is, I'd like to thank you for agreeing to speak at the first Health Optimization Medicine and
Yes.
Practice Symposium on October 20 in Hollywood, Florida. Where you and there's the Microbiome Labs, Novozymes, a conference all there. So it's October 20 to 22. And
Yep.
Kiran, gracious, he said for me he'll do the lecture, and he's going to
Yes.
do a lecture on gut microbiota and health optimization, right? So
Well, and here's the interesting thing about that. I'm actually not even speaking on the microbiome labs part.
oh
I because I decided, I was like, I'll come and attend. I want to see how they put together the program with other speakers and all that. But then, and so I was just coming to attend. But since you asked, I was like, well, I've got to help. I've got to support my friend Ted, and I love what you're doing.
yeah.
It's an honor to be part of it.
So those who come and attend our first symposium during October 20 can go for free to
Yep.
attend the symposium of Microbiome Labs and Novozymes, right?
The microbiome
And,
keynotes.
and,
Yeah.
and those those who are attending Microbiome keynotes are actually free to attend our
Yeah.
symposium. So if you arrive there on October 20th, then please come and attend and see not only me but Kiran. And Kiran,
Yeah.
I cheated here. I got your views on the entire microbiota gut-brain axis. That's going to be my lecture in the keynote for you guys.
Excellent.
See,
Oh, perfect.
so I'm keynoting. So this is an exchange. But then
I
again,
love this. I
yeah.
love this. Well,
So,
I want to say one thing, that I cannot think of a high a more valuable way for a clinician to spend their weekend than dabbling both in the health optimization part of the program and then the microbiome keynotes, right?
mhm,
Because as we just talked about, the microbiome is involved in virtually everything, in
mhm.
health, wellness, sickness, and so on. And Microbiome Keynotes was designed from day one when we put it together as a program to bring microbiome science to clinical practice, right? Bridging that gap between all the academic work that goes on for the science, but then bringing it to clinical practice and giving you tools to be able to take advantage of the microbiome on the very next day, right, with your patients.
Right.
So it's such a practical conference to provide you tools, clinical tools to intervene with your patients, and then you add the health optimization component to it, which is looking at health as a condition, because now not only can you walk away from there understanding how the microbiome impacts wellness and disease processes and how to intervene using the microbiome, but then you can also start to target health in your patients, right? Which very few clinicians do, right? Most clinicians have the view of the absence of disease as health, and so let's just think about disease, right? But not think about health as a condition.
Yeah, and that basically, manipulating your mitochondria to great health actually has not only an effect on longevity, but it has an effect on what I say is the quality of life. As I say, when you ask for long life, don't forget to ask for eternal youth. Where health optimization medicine, we're the eternal youth guys. I'm sure you like that, Kiran.
That's awesome.
So, Kiran, last question. All right.
Yes.
And this was not in the notes that I sent you.
They were not.
No. What is your advice or audience to live? As Prescriptions is the brand of a Smarter, Not Harder, right? The company that I founded to actually fund the nonprofit of health optimization medicine and practice. But what is your advice to our audience in order to live life smarter, not harder?
Yeah, that's a great question. Okay, so number one, I think in order to live life smarter, not harder, is you have to understand how your body works, right? If you don't have basic understanding on what impacts your outcomes, then it becomes a slog of doing lots of things that likely don't have a positive impact. The more knowledge you have, the more efficient you can be, and the more effective you can be at intervening in your own health and then achieving health, actually, right? Which means that programs like this become critically important. Think about the average person and the amount of time they waste on just nonsense, social media nonsense, TV shows and all that. And those have their place, right? I like a good TV show and so on. But if they can invest a little bit more time into understanding their own biology, that will really help them function in a smarter way rather than the harder way of just following 1,001 things, taking 700 supplements and not getting the results that you want to get, right? And I think that's a very, very critical thing. So it's about a time investment into understanding your biology.
Yeah. The way I understand that, I'm sorry for interjecting, Kiran, is that, I have some patients now who say, I ate this and then 2 days later, I got this headache. I tried it again. I ate this. 2 days later, I had the same headache. Even that kind of long term, it's not an immediate thing, but observing, knowing your body, how it's responding to certain things, it requires a certain investment of your time, as Kiran said.
It does, and awareness, right? And so you have to open yourself up to being more aware of what you're putting in, on, and around your body, and how that affects your outcomes.
Right.
And the other thing I would say to people, and I see this as a very common thing that I get when people reach out to me with all the problems that they tend to have, is don't do too many things at once, right? People go nuts. They try 7 new products at once and 4 new devices, and then they're confused as to what may be helping, what's hurting, right? They're like, well, I started this, and this. I went to this conference. I went to the biohacking conference, and I bought 9 new things and 3 pieces of equipment, right? And they're doing all these things. And I'm like, well, how would you ever know what is actually helping or hurting you or doing anything at all if you do all of it at once, right?
Yeah.
So you have to be a little bit more pragmatic about your approach and try things one at a time, maybe two at a time. If you understand that they're complementary or additive in function, and then allow yourself to observe what the outcomes are.
That's a
Right?
patient's issue, right? Do you have
Yeah.
a gut microbiota that will increase someone's patience?
Yeah. Not yet. That would be the Nobel Prize, I think, right?
And your final piece of advice?
So my final piece is knowing that there's absolute hope, right? So there's a lot of people listening to this who are probably dealing with the condition right
Mm-hmm.
now, and they've probably been dealing with it for a long time. They've probably been down lots and lots of Google rabbit holes and trying to understand what this condition is, and maybe they talk to their conventional doctor, and the conventional doctor has said there's no cure for this. It's just something you have to live with, right? Whether it's an autoimmune condition or whether it's a certain type of mood disorder, neurological disorder, like you said, Ted, like fibromyalgia, right?
Mm-hmm.
Chronic pain, things like these that can be very defeating and impact your quality of life. Know that all of these processes have interventions,
Mm-hmm.
right? Unless it's a specific congenital disorder where there's a gene specifically driving it that you can't intervene, and that's very,
Yeah.
very rare,
When it's
right?
anatomic, right?
Anatomic,
Yeah.
anatomical genetic disruption. Those are very rare. The vast majority of things are ecosystem driven, metabolic driven, right?
Mm-hmm.
That can be intervened. So just know that there is hope, right? Don't feel that there's hopelessness. Don't feel defeated. Know that if you keep looking at the right people for the right information, you have to listen to qualified people like Dr. Ted, right? I mean, he's a doctor. He knows what he's talking about. He knows how to vet. He knows how to vet information, right? Versus listening to this TikTok influencer that may look great in a bikini but don't know what the hell they're saying, right?
Well,
Well, you were an underwear model once, I do remember. So you might have had a TikTok page.
you saw that banner, huh?
I did, yes.
That was a long time ago, man.
But those pictures might be out there still.
But I have maintained my abs, so that's good.
Yeah, that's true. See?
And if you see Kiran, guys, Kiran has remained buff and has remained working out, right? Despite
as much as I can.
his crazy travels to back and forth, treating Denmark as if it was just 2 miles over.
Yeah, exactly. Yeah, we do. You have to commit to your body, to your physique. Exercise is one of the foundational things, but know that there's a lot of hope, right? So people will start to feel hopeless, and of course, the definition of depression is really a loss of hope. And know that there is hope. There's interventions. There are things you can do to improve your outcome.
Mm-hmm.
You just have to put in the time. You have to listen to the right resources and learn your biology.
And the willingness to change your lifestyle when you need to.
Yep. Absolutely. You have to make those changes, right?
Okay.
Your behaviors that got you there are not the behaviors that are going to get you beyond it.
That's so
Right?
true.
So,
That's so true.
yeah.
Well, thank you so much, Kiran. This has been, we haven't caught up in a long time. This is our way of
Yes.
catching up. For our audience out there, this is the way Kiran and I catch up. We catch up on
Yep.
the latest papers on almost everything, especially gut microbiota, but on the studies that he's doing. And, where we talk about world peace as if it's already there.
It is within us, at least.
Yes,
Right?
yes. See, and that's the kind of enlightened person that you need leading all of these new ventures. So thank you so much, Kiran, and
It's been my pleasure.
and I'll see you in October. So, guys, we
Yes.
invite you to go see Kiran too, and me. I'll speak, but he's the superstar here as always. And good luck in the new endeavors, Kiran.
Thank you. Thank you. You'll be hearing a lot more about it. I'll keep you updated. And thank you so much for having me. This has been an absolute pleasure and honor. Thank you for putting this program together, making information like this available to people, which is so empowering and so important. And I'm so excited for you to speak at Microbiome Keynotes. That program was designed to provide a platform for people like you to be able to share your brilliance and your knowledge. And I'm honored to be able to speak at the Health Optimization Conference that's going on together with keynotes as well. I think it's an amazing pairing. So really encourage people to go, and I can't wait to promote it if you have any memes or things to promote it, let me know. I want to push
Yes,
it. I want
and
to make sure everyone comes.
we already have the link for ours. We're just waiting for
Awesome.
yours.
All right. I'm going to send some text
Ye-
messages and see if
yes,
I can get that done.
yes. And as to what everything Kiran said about having this information disseminated, this kind of change in perspective and thinking, Kiran and I, when we were at Maria Cristina Hotel in San Sebastian,
Hmm.
looked at each other, and because we were asked why is this information not more widely available, we looked at each other and said the same thing: about time.
Yep,
So it's about time. So
it
thank you
is.
guys. Thank you for listening to this podcast. And thank you again, Kiran, for being in this podcast with me.
Thank you, my friend.
Dr. Kiran Krishnan has explained to us that over the past 2 decades, there have been 5 significant advancements in gut and gut microbiota science and technology. Firstly, the gut microbiota is now considered as an organ, often referred to as the forgotten organ due to its complex ecosystem and vital functions. Secondly, this recognition has led to a shift in understanding of various body systems, such as the gut brain axis, which is now referred to as the enteric microbiota gut brain axis, highlighting the tripartite relationship. Thirdly, the discovery of the gut's enteric nervous system has led to its designation as the second brain and is also now recognized as the body's largest immune system. Fourthly, the discovery of the forbidden interkingdom communication has revealed that outer membrane vesicles, or OMVs, can fuse with human cells, including intestinal cells, and influence their function. Lastly, there have been significant clinical developments, including the creation of tests for gut microbiota, the discovery of its links to various health conditions, and the development of interventions to modulate the gut microbiota, such as probiotics, prebiotics, and fecal microbiota transplantation.